Thank you so much. That's all from me. Yeah, I hope that you guys have a smooth event ahead for these 2 days.
Thank you, ma'am. It was a really interactive video. Professor Suman Jain is in charge of the Brain Stimulation and Neuromodulation Lab in the Department of Physiology, All India Institute of Medical Sciences, New Delhi. She is also the faculty in charge of the Neurophysiology and Nanomedicine Lab. Professor Jain is currently the Vice President of the Indian Academy of Neurosciences, Delhi NCR chapter. Professor Jain has been closely involved with the planning of this symposium from the very inception and has been guiding us as scientific advisor in every aspect of its organization. Professor Jain, we are really looking forward to hearing from you.
Yeah, thank you. Good morning, everyone, all the participants. First of all, I would like to congratulate Professor Manjula Suri, Organizing Secretary, Professor, Department of Physiology and Promotive Health, and Professor Geeta Trilok Kumar, Director, Institute of Home Economics, and the whole team for bagging this prestigious IBRO-APRC Meeting Support Award. Thanks to Professor Pixi Che, Chair IBRO-APRC, for considering the application and awarding this grant. I was going through the scientific program, and then it is actually very well knitted program with all eminent speakers from across various parts of India and also globally. And we have, like Dr. Manjula Suri was saying, there are scientific lectures starting from basic neurosciences, covering pain, inflammation, neurodegenerative disorders, moving to the recent advances, recent therapies like stem cell therapies or maybe transcranial magnetic stimulation, and then some recent very hot topics like gut-brain axis and how it can result in generation leading to neurodegenerative disorders. So, and there was a very good response, like they're saying there are around 200 participants involving not only the students, but there are young investigator, faculty members, and also senior faculty members. So I'm sure that this symposium will be able to motivate all the young and not so young investigators and students to pursue their research career in neurosciences. Akin to IBRO, which Professor Pixi She was showing us about the IBRO, in India, we also have a very huge neuroscience academy, which is Indian Academy of Neurosciences, which came into existence in 1982 by a group of senior neuroscientists, just to name a few: Professor Bhargava, Professor Mehndi Hassan, Professor Parmar, Dhawan, Dr. Dhawan, Dr. P.K. Seth. They initiated this neuroscience academy, and this academy has been very active in promoting neurosciences in various parts of India. And it has been holding meetings regularly at different centers. The last meeting was held at Kolkata, ISER Kolkata, and before that we also had at Hyderabad. Then in 2019, AIMS Delhi, it was this annual conference was held. Besides that, we have regular conferences. The Indian Academy of Neurosciences also promotes organization of workshops or symposias or lectures. More recently, in 2019, we have also started the Delhi NCR chapter of Indian Academy of Neuroscience, because we realized that there are very few labs in Delhi or across Delhi which are actually which are dedicatedly working on neuroscience or neurosciences, and therefore to be able to promote or motivate youngsters to take up research, take up neurosciences or neuroscience field as their career, we have initiated this Delhi NCR chapter, wherein we have some grants also, and so and regularly we're holding training workshops also. So in May 2021, a series of workshops focusing on virtual workshops focusing on very specific techniques involved in neurosciences were showcased by the eminent neuroscientists from Delhi NCR chapter. And then we had few lectures, and therefore to bring all the neurosciences together in India, and as well as like that is to expose them to the global opportunities which are available. This Neuroscience Academy is working on that. And for neuroscientists to be able to attend various international conferences or international workshops which are organized by various societies like IBRO or SFN or other type of other societies which have dedicatedly neuroscience related activities. We also have travel fellowships to the candidates. They can apply for various fellowships, and then they are able to attend those conferences. So Indian Academy of Neurosciences also have very good amalgamation of basic and clinical researchers, and also it has neuroscientists from various other countries involving Germany, Japan, Poland, Saudi Arabia, Sri Lanka, Africa, UK, and USA. Currently, Professor Ishan Pantro from Jivaji University, Gwalior, who's the leading and eminent neuroscientist, is the president of Academy. And so as Vice President of Indian Academy of Neurosciences, I also take this platform to request all the youngsters and even the senior investigators to please join this academy so as to form a neuroscience circle and to promote neurosciences in India as well as in so that you get exposed to various agencies or various organizations globally. So this will be very helpful for your enhancement of your career, or maybe you'll be enriching your neurosciences social circle, as we call it. I will not take much time. I would just say once again, I would like to congratulate the whole team and convey my best wishes for the grand success of this symposium. I'm indeed privileged to be a part of this symposium. Thanks, Dr. Manjula, and best of luck and happy learning to all the delegates. Thank you.
Thank you, ma'am. Thank you so much. So we'll be beginning the proceedings for today by Professor Fusao Koto, who will be joining us at 9:40 a.m. sharp. So I request you all to please be patient and let's see Professor Kato in some time. So we can have a break for 10 minutes and rejoin sharp at 9:40, please, if you could. Thank you. Thank you so much for your patience. So we'll begin today's proceedings with the first presentation by Professor Fusao Kato. Professor Kato is a medical doctor with a doctorate in pharmaceutical science. He's also a professor in the Department of Neuroscience and director, Center for Neuroscience of Pain at Jukai University School of Medicine, and a member of Science Council of Japan. Professor Kato's research areas include synaptic plasticity involving amygdala. He has examined the role of the central amygdala in widespread sensitization seen in chronic pain. His presentation today is titled Central Amygdala in Nociplastic Pain. Professor Kato, we are very honored to have you join us today, and we really look forward to listening to your informative presentation. Professor Kato.
Thank you very much. Good morning. Can you hear me or not?
Yes, we can hear you.
Okay. Good morning. This is afternoon in Tokyo. And how are you? How?
Thank you.
As a matter of fact, it is snowing in Tokyo now. So today it is very cold. So thank you very much for providing me such an occasion and such an honor to speak to you. And I have been many IBRO neuroscience school in many countries, including Nepal, including Iran, and many other Asian countries. And I have been there in India 3 years ago, before the COVID-19. I visited Chandigarh, and I had a very nice time there. So I have a very good memory of visiting India. And so, of course, visit India in the real world, not through Zoom and teleconference. But no, it is yet difficult to visit India now, but I will enjoy speaking to you. And it is my great occasion for communicating with you, the people in India, also from all over the Asian countries. Okay, so I'm now trying to put my PowerPoint on the screen. Would you give me some more time, because my PowerPoint file is not yet downloaded now. This will be all right.
Yeah, you can take your time. We have stopped sharing our screen.
Thank you very much. So how is the situation there in India, the COVID-19?
As of now, it's under control. Currently, there is no spike. It's not increasing as of now.
Okay, yeah. Yeah, this is also the case in Japan. You know, the number is decreasing every day. And we have most of us received the third dose of the vaccine. I don't know what happened to my PowerPoint file. Okay. Go.
Professor Kato, if there is some problem, then we can present it for you.
Yes, okay. I think I'm ready. So would you wait a moment for
Sure.
sharing my screen? Yeah. I'm sorry, my PowerPoint had some problem, but now it is okay. Hello, can you see my screen?
Yes, we can see it. We can see it. It's visible.
You can see the big Asia map.
Yes, sir.
That's good. That's good. So I can start now. Okay.
Thank you.
So I'm sorry to have had some time to prepare and project this screen to you. And I hope you can see now everything. Okay, no problem. So thank you very much for the kind introduction. And my name is Fusao Kato. So I'm in Tokyo here. This is Japan. And a few days ago, we had a very strong earthquake. And I was quite near to that center of that earthquake. And so everything is stopped and train. We have a bullet train and it has stopped. And still there are some fire electric generators are still out of function, out of order, and so they are trying to prepare them, but repair them. But still, we are facing the lack of the electricity. So this morning, the government of Japan said to the people that you have to turn off the heating and electricity, everything. You must turn off your television and everything. So I think there's a deficiency of the electricity today, and still it is very cold outside and it is snowing. So it is a very hard time for us to Japan. And so I'm connecting from here to you, to India here. Most of you are in New Delhi, I suppose. And so this is the city of Tokyo. So this is from here. My building here is here. I'm staying here on the 11th floor of this building. You can see the Tokyo Tower here, and this is the Bay Area, and you can see the bridge here. And I'm talking to you from this building now. Okay. So this is the disclosures. So today I'd like to talk to you in two topics. So first part will be the backgrounds, and second part will be the mostly with our data, recent data, that might explain to you that's the role of the central amygdala in nonsplastic pain model. So I think this is neuroscience meeting, neuroscience school. So I'm not so sure whether you are very familiar with the science of pain. So I'd like to start with the science of pain, the definition or meaning of the pain, okay? So what is pain? Do you know what is pain? Can you explain? Can you try to explain what the pain is? So pain is maybe something that you have, all of you have ever experienced. You know what pain is. So we know what pain is since very early of life. We don't know when it was the first time that we learned it, but we know what pain is. We understand, we learn what the pain is through the life from the very early part of the life. And we understand that pain is necessary for the survival. So it has some very strong role of avoiding injury or everything, or burning everything. But there are some patients, human patients, of course, human patients without pain. So the name of the disease is called hereditary sensory and autonomic neuropathy, HANS. And these are the patients. For example, we have in Japan, we have about 200 patients like this, and they suffer from severe tissue damage since very early of life. And even the evolutionary old invertebrates, such as octopus, or Drosophila, or fish, of course. Fish are vertebrates, but all the invertebrates like octopus, they have nociceptors. They have injury-associated plasticity, and they have network for nociceptive processing, and also a pain-associated learning system. Okay, so this is highly conserved system, and it is an alarm to ensure the survival of individual animal, individual body, and species also. Thus, evolutionary conserved, rather it is an essential function for evolutionary selection. So pain is something related to some to protect your body and species, everything. Okay, so this is quite important and essential function. So there are 4 essential biological functions of pain. So 1, escape and avoid. Number 2, protect. Number 3, prioritize. Number 4, promote learning. There are essential 4 important functions of the pain. I would like to explain for you. For example, if you touch something very, very hot, you withdraw your hand. Okay, when you put your feet on on a nail or a pin or something like that, you will withdraw your feet. So this is the primary function of the pain. You should escape or avoid these touching some hot material or very hot stove will cause you burning. So immediate escape from ongoing tissue damaging events. This is the primary role of the pain. And second, protect. So if you have some fracture of your bone here, or the joint here, and it's better to avoid moving that joint, okay? And so this means that, when you move this joint after a fracture or injury, that has risk to delay or to make it impossible to have complete recovery. So this means that, it is better, much better to avoid moving around or using these joints and feet, everything that. So this, you have to be very, very sensitive to moving that joint. So in that case, this pain has a protective function. So it facilitates the recovery of damaged tissue or by protecting it from further injury. So the function called is sensitization or hypersensitization of your sensory system, makes you protect from the further injury. The third function is prioritize. So, for example, imagine that you have a headache or you have a stomach ache, okay? And if you have headache and listening to my talk, and you will never be able to concentrate on my talk, okay? So if you're getting an exam of something, mathematics or whatever, and if you have some very strong stomach ache, for example, and you cannot concentrate to solve your questions. So this means that the pain gives you the priority to the pain. So the pain tells you that you have to solve this problem. You have a headache, you have a stomach ache, you have to solve your problem of head or stomach. It's not the time to solve your questions, pass your exam. Okay, so this is one of the functions of pain, which is called prioritize. And the fourth function of the pain is promote learning. So, for example, if you have some injury on the feet or something there, and you will learn to avoid that situation. So the most famous case is the conditioned fear learning or conditioned threat learning. So in that case, when you have some mild pain or some nociception, that will allow you to avoid from that situation in the future. Okay, so this is something the pain tells you to learn, memorize this situation to avoid the same injury in the future. So this is a function which is called promote learning. So they have these varieties of biological functions, and the pain is quite essential for survival. So the central question of pain biology is how these functions are realized by the brain network, and how dysfunction of this system leads to aberrant, often chronic pain. Okay, so this is the fundamental idea of the pain studies. So next question is, what is pain? We have to define what pain is. Would you please try to define what pain is? After our experience, pain should be something caused by some source or some cause. So this means that some injury. So injury is on your hand, and the hands send the signal to your brain, and pain is there. Okay, so this is very classical and easily understandable structure. But this is not the case. This is not always the case. So there is an association, society called the International Association of Study of Pain, and which defines the pain like an unpleasant sensory and emotional experience associated with or resembling that associated with actual or potential tissue damage. This is the official definition of pain. So there are many aspects and important factors in this definition. So first, it says that it is always unpleasant. And it says also it is sensory and emotional. It is not only the sensation. You might think that pain is a sensation, but it's not the sensation only. It's sensory and emotion. It has always negative, unpleasant emotion, okay? And this is experience. So this is not the sensation. This is not the response. This is not a physiological response, but this is experience. So this means that it is quite subjective. And it is quite interesting, but because this definition tells that it could be something that resembles that associated with potential tissue damage. So resemble means that if it seems like pain, it is pain. For example, with this definition, we can define the pain in animal, because the animals, they don't say, I feel painful, it is painful, I feel so painful. They don't say that. So you can define the pain in animal, newborn baby, who doesn't say anything, and vegetative state patients, so they can't say a word, they cannot explain themselves, they express themselves. But if there's some responses resembling to the pain patients, you can say that they have pain. And finally, it is defined that it is associated with actual or potential tissue damage, or just resembling to that. So this means that actual tissue damage is not essential for pain. So this is quite interesting, because as I told you that, you might imagine that pain occurs from some cause or source like injury or the damage of the tissue, and the result is pain. But this is not the case. So let's forget this idea of causality of pain. So pain can occur even in the absence of the origin or cause. So there is not, following this definition, that it is unquestionably a sensation and a parts of the body, but it is also always unpleasant and therefore also an emotional experience. And pain is always subjective. So you might imagine how difficult it is to scientifically scientifically solve this problem of pain, because it is always subjective. And there's a word, noception. So you can see this image, noception here. Noception is something that the damage, the detection of the damage of the tissue, detection or transduction of these inflammatory events or damage, injury, everything. So this is called process. Noception, and noception is not a pain. Pain is something comes after. So if you, this is a schema drawn by René Descartes about 400 years ago. So it explains that if there is a noception, the signal is sent to the brain, and the brain feels a pain. So this is the schema which was conceived 400 years ago, but as I told you, we have now a lot of cases that pain occurs even without the noception, or pain, the intensity of the pain has nothing to do with the intensity of the noception. So we should forget this, and we should change to a schema much complicated, using on the basis of a more recent advance in the pain physiology. So if there's a noxious stimulus that damages the tissue, this signal is sent to the brain, and the prior experiences, attention, expectation, mood, anxiety, depression, that affects the pain, intensity of pain, neurochemical and structure changes, genetics, and sensitization occurs, which is regulated by the brain. Okay, so we have to change our view of pain. There's injury, and the result is pain is not anymore justifiable. Okay. One of the reasons why is about after 1991, many people have imaged the brain, the activity of the brain, and how or where of the brain is activated during one is feeling pain. So the people have found that there are a lot of places, that regions in the brain are activated. These are the pain metrics, which are the structures that are excited during pain. So there are a lot. You can see, of course, there are the thalamus, and of course there is a sensory cortex. But in addition to that, many anti-cingulate cortex or amygdala or cerebellum, many other structures are activated during the pain. Widely dispersed brain areas are co-activated. Many of these have strong link with emotion. So it is quite normal, because pain is an emotional experience, and there is no single pain center. And parallel co-activation of these structures forming this matrix results in various types of pain. So this is what we experience in the pain. So the brain, many brain regions are activated. So let's move to the next topic, because I generally talk about the pain, but now I want to focus on the chronic pain. Okay, so chronic pain. Chronic means that persistent, long time, sustained. Okay, but it's not just long-lasting pain. It is a disease. So chronic pain is a serious medical concern. The definition of chronic pain is persistent or recurrent pain for at least 3 months. So 3 months is the criteria for the chronic pain. And it affects more than 20% of the populations in developed countries. The social loss is huge due to the significantly lower quality of life, a risk factor for mood and emotional disorders. In some patients, the primary cause can be identified, but in other patients, the primary cause of the pain cannot be identified. So in the cases like central neuropathic pain, stroke pain, phantom limb pain, and fibromyalgia. And in many cases, unfortunately, conventional analgesics like NSAIDs and opioids are ineffective, and there is no gold standard drug. And very often chronic pain is accompanied by various types of plastic changes in the brain pain matrix. So it is a kind of, one might say, it is a kind of a neuroplasticity disease. I'll come back to this point later again. And in 2018, so just 4 years ago, WHO has defined has issued an international classification of disease version edition 11. And for the first time in the history, they have described chronic pain as a disease. So now, from since 4 years ago, chronic pain is not a type of pain, but it is a disease. Please remember this. And so pain is classified into two parts: chronic pain and acute pain. Acute pain is pain caused by the ongoing injury or something, and chronic pain is divided like this: chronic primary pain, cancer-related, post-surgical, secondary musculoskeletal, secondary visceral, blah, 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 blah. And the most important and the most exciting classification of this chronic pain is chronic primary pain. Primary, what does it mean primary? Primary pain means that pain is the primary. So this is not a result, this is not a secondary consequence of something, but pain is the primary. So chronic primary pain is chronic pain. And in one or more anatomical regions that is characterized by significant emotional distress or functional disability. And chronic primary pain is multifactorial, biological, sometimes psychological, sometimes social factors. These factors contribute to the pain syndrome. So this is quite complicated, and it is quite normal because, as you have seen, that many brain regions are involved in the sensation of pain. So this means that chronic primary pain is purely caused by the brain activity. And recently, International Association of Society Study of Pain have proposed that there are three mechanistic descriptors, three mechanisms for chronic pain. So the first one is nociceptive pain. So if there is some injury, nociceptors detect that injury or some inflammation, it is detected by nociceptive nerves, the nociceptive fibers, and this is transmitted to the brain and it causes pain. So pain caused by nociceptive activation. I think that most of these secondary musculoskeletal pain, secondary visceral pain, this post-surgical pain, this is kind of a nociceptive pain. There is some injuries, there is some activation of nociceptors. And second class of chronic pain is neuropathic pain. So there is no injury on the site. You feel pain on the hands, on the feet, on the knee, on the shoulder. But pain is caused by disease or injury in the somatosensory nervous system. So this is neuropathic pain. So this classification here in this chronic neuropathic pain here, okay, this is neuropathic pain, so this is neuropathic pain. But even with this first class of nociceptive pain and second class of neuropathic pain, there are so many patients who complain pain, who suffer from pain without nociception or without neuropathy. So IASP took many years to discuss and to classify this third group of the mechanistic descriptor of pain. So this is what we call nociplastic pain. So this word is introduced by ISP in 2017, so just 5 years ago. And the definition is pain arising from the altered function of pain-related sensory pathways. So this means that plasticity, this is a kind of a brain plasticity or neuroplasticity. And so pain arising from the altered function of pain-related sensory pathways in the periphery and CNS, in the brain, of course, and causing increased sensitivity with that activation of nociceptors and neuropathy. So it isn't necessary to have some nociceptor activation. It is not necessary to have neuropathy on the sensory nervous system, but just the aberrant brain activity can trigger the sustained, long-lasting pain, which is called nociplastic pain, and which is based on some plastic change in the nervous system, particularly in the brain, okay? So I'd like to explain a little bit on this nociplastic pain. The definition of this nociplastic pain is pain arising from the altered function of pain-related sensory pathways in the periphery and CNS, causing increased sensitivity in the absence of evidence for activation of nociceptors and neuropathy. This is a concept and definition introduced to describe patients claim pain, but without any demonstrable tissue damage, injury, or injury of sensory nervous system. So this is a very new class, but the patient, the medical doctors or pain specialists have known from long ago that there is this type of pain. Possible patients with nociplastic pain include chronic widespread pain, of which I am going to talk about more in detail using the animal models, and the fibromyalgia, chronic low back pain of unknown causes, chronic temporomandibular pain or disorders, irritable bowel syndrome, chronic primary bladder pain, or chronic primary pelvic pain syndrome. There are so many people, so many patients, about 7 to 10% of the people are suffering from this kind of pain. And they were diagnosed that they don't have any problem, they don't have any tissue damage, they don't have injury, inflammation. So the medicine today cannot do anything for you. So this was the responses of the doctors to this kind of patients. This used to be. Now we have definition of nonspecific pain, so these patients can be classified as nonspecific pain patients. So now it is the time to develop drugs against nonspecific pain. So, and what is complicated is nonspecific pain is often accompanied by fatigue or sleep disturbance, mood and memory problems. So it is a kind of, psychiatric disorder. And in the psychiatry, there is also a very similar definition of the pain syndrome, which is called somatic symptom syndrome. But this is quite similar. And in these patients, they very often shows central sensitization. Central sensitization is sensitization of the skin or body parts, which is caused by brain or central nervous system. And many studies have described that there is plastic changes in the brain in the chronic pain patients. So this is an example of the study which was published several years ago by the group of the Professor Apkarian in Chicago, and they have followed the patient number of patients, and they made every 3 months this kind of functional imaging of the brain, and these are the images of the patients which had persistent pain even after visit 4. Visit 4 is 1 year after. And these are the brain images of the patients with chronic back pain. But 1 year after, this back pain is recovered. They don't suffer anymore. And you can see the quite difference in between the 1 year after images between these persisting patients and recovered patients. The recovered patients, they don't have any significantly different activities. And so this is a change in the brain activities. So this is in pain, emotional, and reward systems. So the people who have persistent low back pain, they have some changes during, this is week, so this is 1 year, different time course of these changes. And this is the amygdala and medial prefrontal cortex. So the activity in the amygdala increases in 1 year after, and in the patients who had recovery of the low back pain, these activities goes to almost normal level. So there's some limbic system, including the amygdala, plays a very important role for this persistent pain. So they follow up these patients up to 3 years, and after 3 years, they have analyzed what is the cause, what is the factor that might distinguish between the recovery group and the persistent group. And so they have identified some gene. This is kappa opioid receptor, and they have identified also the amygdala, volume of the amygdala, and the functional connectivity between the amygdala and accumbens and PFC, and this is also related to the white matter size here. So they have concluded that the limbic system, including the amygdala, plays an important role for the persistence of that chronic pain. So this is in the human patients. So the amygdala in chronic pain. So this is the reason why I think that the amygdala is important. What is the role of the amygdala, or how the amygdala is involved in the pain? So let me start with this one. So this schema I took from the latest version of the Kandel's Principles of Neuroscience. Maybe you have already have this book, and the chapter of the pain. They have described this schema. So it starts with the nocturnal stimulus. They form synapses here in the spinal cord, dorsal horn, and they go up to the pons. And the first synapse they form is one is parabrachial nucleus, and the other one is at the thalamus. And this is quite conventionally well-known thalamus, and they project to the primary somatosensory cortex. It follows all the rule of the other sensory system, thalamus cortex. Okay, but importantly, they have described the spinal cord to parabrachial nucleus here. And from the parabrachial nucleus, there is a direct connection, projection to the central amygdala. So this pathway is bypassing the cortical systems, and this is the major targets of the nociceptive projecting neurons in the trigeminal spinal cord and the dorsal horn, the very superficial layer of the dorsal horn. So this means that nociceptive information primary target to the parabrachydactyl nucleus and the central amygdala. So this is the reason why amygdala is quite important. So the people has called this system spinotrigeminal parabrachial amygdala system. It starts from that this periphery, like here. This is for the spinal nerve. This is for the facial nerve, and this is trigeminal nerves. Oh, I'm sorry, this is not facial. This is trigeminal nerve, facial part of the body. And this is trigeminal, so they go up to here, and they go these neurons, this nociceptive primary afferents project to dorsal horn, and from dorsal horn, there is a projection to the lateral parabrachydactyl nucleus, and this lateral parabrachydactyl nucleus projects to the central amygdala. You might think that the most important projection is the thalamus, but this is not the case. You know, the people have identified, or at least in the rat and mice, almost 95% of the projecting neurons from dorsal horn or spinal trigeminal nucleus, they project to the lateral parabrachydactyl nucleus. So lateral parabrachydactyl nucleus is the major, most important target of nociceptive neurons, nociceptive projecting neurons in the spinal cord and the trigeminal nucleus. Okay, so this is lateral parabrachydactyl nucleus, and this lateral parabrachydactyl nucleus then project monosynaptically to the central amygdala. Central amygdala is here in the limbic system. Okay, and this pathway conserves only weak somatosophy. This means that they don't care which part of the body this signal, nociceptive signal, comes from. So they handle all this information arising from multiple parts of the body, and this pathway allows the amygdala to collect information as to anything wrong, something wrong is happening in my face and body. Okay, so this is really danger and alarm related function. These pathways plays important function, of course, in the pain. Myself, I have been interested in this system, and I have been working on this system. And since early 2000, we have published many papers describing there is a neuroplasticity in this system. Okay. So, for example, in the neuropathic pain models of the rat, this pathway, this synaptic transmission here from here to here is potentiated in a manner dependent on peripheral C fibers. And we have also demonstrated that inflammatory pain can cause this potentiation, and it is completely dependent on the CGRP, calcitonin G-related peptides. So if you knock out this CGRP peptide, this potentiation does not occur. And optogenetic stimulation of this pathway here serves as non-conditional stimulation for auditory associated threat conditioning. So as I told you, this is learning function of the pain. So if you just stimulate this pathway optogenetically, artificially, without very nociceptive information, you can learn in adversarial situation. And also we have published some chemogenetic modification of the CA neurons. And also we have done MRI in the small animal MRI we have, that is 9.4 Tesla. And we have identified that if you inhibit the central amygdala, CEA, with chemogenetic techniques, you can have the very big change, big decrease in the activity of the limbic system. So I have finished the part one. The amygdala is quite important in the chronic pain. You now understand what is pain, and you now understand what is chronic pain. And there are some specific form of chronic pain, which is primary chronic pain, or in other words, nociplastic pain. So let's go to the nociplastic pain. The part two is central amygdala in nociplastic pain model. Okay. So the thing that we have done is we injected formalin. Formalin triggers very strong inflammatory responses. So we injected formalin to the face. So if you look at this video, they are rubbing their face because there is some irritating in the face. And if you observe this activity, this is time, and this is time in minutes. So you can have this kind of rubbing behavior, but this behavior terminates in 1 hour. So after that, they don't feel anything. They don't show any sign of some inflammatory disturbance on the face. Okay, so this is some latent inflammatory pain, and afterwards we are interested in what happens afterwards. Okay, and because there are some reports showing that after formalin injection, after this early nociceptive behaviors, there is an increase in the C-phos, there is an increase in the P-ARK, phosphorylated ERK, there is an increase in the SRC, MAP kinase, and everything, COX-1, everything. And finally, they show neuropathic pain like pharmacological behavioral phenotypes. So we are interested because one injection of formalin onto the face, what it would be the consequence of this. So what we did was first we observed the parabrachial nucleus, which is the direct, most important target of the trigeminal or spinal, but in this case, trigeminal primary afferents. So they project to the right parabrachial nucleus. We inject the formalin to the left side of the face, and this is left injection. And there is some slight difference, but left parabrachial nucleus showed a larger number of FOS. FOS is a molecule which is immediately activated or expressed by some neural activities and related to some plasticity. And if you inject to the right side of the face, they show some increase in the face, like on the left, right side of the face, but both sides are increased like this. So this is significant increase of the number of the phosphopathogenic neurons. So we climbed up to the central amygdala like this, and we counted the central amygdala, what kind of activation occurs in the central amygdala after formalin injection. So this is 3 hours after the formalin injection. And if you inject the formalin to the left side of the face, we could see the right side of the amygdala is activated. So this is the statistics summary. So you have a nice significant increase in the positive neurons in the right central amygdala. And then what we did was we injected formalin to the right side of the face, and what I have observed was again increase in the formalin in the right side of the face. So we were very surprised. We repeated this experiment, but we are very, very confident that only the right side of the amygdala is activated regardless of the side of the injection or inflammation. So this was surprising. Right side of the amygdala should play some specific role in this pain-related some plastic change later. So this was the activation of parabrachial central amygdala system in latent inflammatory pain. And as you have seen that this there is some specific laterality. Right side of the amygdala plays more important role in this activation. So what would happen in the synaptic potentiation, because the FOS is involved in the many plastic processes. So we tried to analyze transmission. So we made slices and we made electrophysiological recordings. We recorded from central amygdala neurons, and we stimulated this pathway, and we measured the excitatory synaptic potential current, and because they are the glutamatergic, so there is a release of glutamate from the terminal, and there's an activation of AMPA receptors. So this is AMPA receptor mediated current. So in this case, this is control saline. So this is an injection to the left side of the face, and this is the stimulation intensity and EPSG amplitude relationship. So if you increase the stimulation intensity, you will have more number of fibers firing, and so you can see the larger amplitude of EPSG because the number of glutamate is increasing. And we compared with this formalin injected animals, so the amplitude. So this means that there are larger number of glutamate receptors are released, glutamate are released, and larger number of glutamate receptors are activated. And this is the case of the left injection of formalin, and this is the case of the right injection of the formalin. And this bold line shows the right side of the amygdala. So this is also the case for this right side. So you can compare here and here. Always the right side of the amygdala shows a larger amplitude, much larger amplitude compared to the left side. And this is the right side amygdala. So you can see at the same intensity, you can see it twice or third, 3 times larger amplitude only in the right side of the amygdala who received the formalin. And this is not the case with the left side amygdala, and this is not the case for the saline injected animals. So this means that there's some specific and very robust and strong potentiation, synaptic potentiation in this LPB2CA synapses. Okay. So I have shown you that there is a synaptic potentiation of parabrachial central amygdala synapse in latent inflammatory pain. So the next question is, what is the meaning of this potentiation? If your microphone is on, please turn on it. If you please mute it because I can hear your voices. Okay. If you're not muted, please mute your microphone. So what we did was we used this model. So they show this rubbing behavior for 1 hour. And after that, they don't show any specific behavior. So after injecting formalin to the face, by chance, we tried to analyze the sensitivity at the hind limbs. So we made the foam fry filament test. So if you touch the hind limb like this, and so we measured the threshold for this escape behavior, withdrawal behavior. And surprisingly, what we have seen is if you inject formalin to the face, not the feet, I mean, if you inject formalin to the feet, the sensitivity to touch and the bilateral hind limb is decreased. So threshold is decreased. So it means that sensitivity is increased. Yeah, okay, this is drastic. And this is 1 hour after the formalin injection, 3 hours, 4 hours. And this is 3 days, 5 days, 7 days. So we could observe this lowering of threshold. In other words, sensitization of the hind limb, even after 1 single shot of formalin to the face for more than 13 days. So this means that, this inflammation only once and short-lasting inflammation cause long-lasting sensitization in the hind limb. So we thought that this is kind of widespread pain. Widespread pain is described very often in the patients' pain experienced at remote and distributed body regions. This is a hallmark symptom for chronic pain, particularly associated with primary chronic pain, such as fibromyalgia. And in some cases, it is associated with injury or inflammation at the distant site, such as orofacial regions for the pain in the limbs. And for this reason, it is often underdiagnosed because, if you have some pain in the hand and your doctor doesn't examine your feet. If you have some pain in the face, your doctor doesn't examine your hands. So this is the reason why it is often underdiagnosed. And for example, the dentist do not usually examine the pain in the forearm or trunk. Okay, it must be very strange. And no injury or no inflammation at the site of pain. So if you feel pain in the hands and there is no injury or inflammation there, so it is highly likely the central sensitization through nociplastic mechanism underlying it. You can understand now. Okay, so this is widespread pain. So we thought that this hypersensitization in the hind limb of the rat, where there is no injury, could be a good model for widespread pain. So we asked the question, what is happening here? And so what we did was, what we used was a DREAD technique. DREAD is a designer receptor exclusively acted by designer drugs. So we transfected the AAV vector and this DREAD receptor, which is activated only by artificial ligand, which is called a CNO, into the right side of the central amygdala. And so we measured the same thing. We injected formalin, and so there is a decrease of the threshold. And here we injected artificial ligand for this DREAD receptor. So what happened here was there was a recovery of sensitization here at the right side and the left side of the hind limb. So this means that if you inhibit central amygdala neurons, this sensitization is recovered. And quite interestingly, this very strong recovery could be observed when you inject this vector into the right side of the central amygdala. But if you inhibit the activity of left side of the amygdala, nothing happens. So again, here we are confirmed that right side of the amygdala should be involved in some pain regulation in this case. So to summarize here, the interpretation of this is right central amygdala neurons activated by facial inflammation affect descending pain modulatory system and enhance tactile sensitivity to cause hand limb sensitization. This is the interpretation. Therefore, inhibition of the right side of amygdala neurons by inhibitory dread that we have shown you attenuated this inflammation associated hand limb sensitization. So this means that this kind of latent sensitization, the central amygdala plays an important role after the inflammation on the face. So next thing, the last thing that I'm going to show you is the question whether what kind of things happen if you artificially activate the right side of amygdala in the animals without injury, without inflammation. Just activate the right central amygdala. What happens if one artificially excites central amygdala neurons in animals without inflammation or injury? Will it cause tactile allodynia? So this is the question. So we transfected this excitatory dread HM3DQ into the central amygdala. So we made a slice like this after the experiments, and we have confirmed that CNO, this artificial ligand, can excite the neurons which express these vectors. So this receptor, okay? So we observed the behavior. So in this case, we didn't inject the formalin. We just injected saline, okay? So they don't show any reduction of the threshold. So they are not sensitized. And this is right hindpaw, and this is left hindpaw. But as soon as you inject CNO to this animal to excite the central amygdala neurons, what we have seen is huge decrease in the threshold. This means that even just single activation of the central amygdala neurons on the right side of the central amygdala, you have a very strong sensitization both in the right hindpaw and left hindpaw. So this was very, very striking. And this happened only if you excite the central amygdala neurons in the right side. If you excite the left side central amygdala, nothing happened again, like this, okay? So this is a summary of the results. So if you have some orofacial inflammation, the right side of the central amygdala is activated, and there is a synaptic potentiation. So you have some neuron and synapses activated. And in that animals, there is ectopic tactile allodynia in the bilateral hind limb. And if you inhibit these right side amygdala neurons with DREAD technique, the hind limb allodynia is attenuated by simply inhibiting the right side of the amygdala. And if you excite the central right side of the central amygdala in non-inflamed, non-injured rats, it causes de novo tactile allodynia. So this is a summary of the results. The conclusion of this series of experiments is the right central amygdala controls the tactile sensitivity in widespread border regions in response to ongoing or potentially harmful situations. So this is the role of central amygdala that controls this non-spastic or primary chronic pain. So this is a take-home message. The central amygdala actively controls tactile sensitivity in widespread multiple body regions. And we know that as the central amygdala neurons are activated in ongoing aversive situations as body, such as inflammation, injury, stress, this system might contribute to protecting the body against these harmful events of further injury or attack. Thus, this augmented tactile sensitivity might form a kernel of the vigilance, defense, and survival function of the central amygdala, and likely to be phylogenetically conserved. And this function might also form the basis for primary chronic pain. In other words, non-spastic pain, such as fibromyalgia, with widespread pain in human patients. So which might be a very important, a very good target of the drugs to be developed in order to attack these non-spastic pain patients. So these are the people who are involved in the data or acquisition and analysis and writing papers. And so I should thank all these people, and I should thank also the funding from these agents. So thank you very much for your listening.
Thank you, Dr. Fuzakato, for this excellent, informative presentation. And we are really honored to have you with us. And I would like to ask one or two questions. Especially, I would like to know what is the, or is there a difference between the chronic sensitization, nociplastic pain, hyperalgesia, allodynia? Is there a difference between these terms?
I should say that there is no difference, but okay. There are some, there are some situations, or there are some phenomena, there are physiological responses, they belong to different grade of the descriptions. So in some cases you can have hypersensitization in nociplastic pain models, in central sensitization. So they are name of the phenomena, or the name of the classification, the name of the phenomena of some subtype of the pain. So just they present some distinct aspects of the whole phenomenon, and so some patients could have two different types, like allodynia and sensitization, but some other patients could show some other phenotype, but they are only a part of whole system. And the largest classification is primary chronic pain. This is the largest, and nociplastic pain is a kind of classification within. And allodynia and hypersensitization, they are some physiological phenomenon observed in these type of pain situations.
Thank you.
Is it clear?
Another thing we've noticed in COVID, since pandemic is still going on, although the incidence is lower now, is patients complaining of central sensitization and nociplastic pain. If you have any comment on that?
Yes, we have a lot of examples for that, and it is quite interesting. But you might know that, for example, vaccination. If you have vaccination against COVID-19, you have many side effects, undesired side effects. But this side effect is not always with your arm where you injected your vaccine. It comes sometimes in your back, low back, or sometimes in other parts, or sometimes headache. So this means that, this vaccination and activation of inflammatory systems might activate the central nervous system, that makes the protection level more higher, and that might result in some other parts of the body's sensitization. So this is one of the things. And another thing is, there is a paper showing that a large part of this COVID-19 vaccination side responses are caused by nocebo responses. So there is a placebo, and there is also nocebo. So nocebo is, if you are told that this vaccination or this injection will be harmful, that will make harm, very, very painful, and they are convinced of that idea. So people are concentrated on your arm or your low back, like this, and they are more easily to feel pain in the back or arm or whatever, headache or anything. So this is kind of nocebo effect. So this is in contrast to the placebo effect, as you know. Placebo is, if you inject anything, that might be effective in reducing pain. So this completely opposite side of the effect. But there are a lot of nocebo-like responses with this vaccination, because people and the newspapers, television tell that is harmful. Very many people feel pain in the hand, the head, and so this might affect the many patients brain activity to concentrate onto the pain in the body parts, and that might augment the incidence of some side effects with pain. So in very very wide meaning. This is the kind of the plastic or central sensitization problems with this COVID-19 pandemic that we have experienced.
Okay. There's one question in the chat box.
Sir, can you highlight the role of medial PFC in pain apart from main role of amygdala?
Yes, important things. Yes, there are many papers showing that medial PFC plays an important role. But when you think of pain, or when you discuss the pain mechanism or pain circuit network, you must always remember that all these networks are strongly connected. Medial PFC have very strong projection to the basolateral amygdala, and basolateral amygdala project very, very strongly to the central amygdala. And also central amygdala project to the accumbens nucleus and also to the cingulate cortex. So they are completely mutually connected. And it is for us, for the researchers, it is quite difficult to understand what is going on because, it is not an element, but the network is behaving differently in these chronic pain situations. And there are some lines of evidence showing that MPFC plays an important role of controlling all these central amygdala responses and PFC ACC responses. So this means that you might think that PFC is playing an important primary role, but no, it's not the case, because PFC is also receiving various nociception related influences. So their activities are mutually regulated. But quite importantly, MPFC is very, very well developed in the human beings. So people think that MPFC might play important roles in this kind of placebo effect or some very psychosocial effects of this sensitization or persistent pain symptoms with nonsplastic aspects. And so in that case, in the human beings, the importance of MPFC is quite well demonstrated.
There is another question by Manish Asthana. He has asked, what is happening after 20 days? It seems pain returns. So you showed a graph where you plotted the pain versus days. I think he's asking about that. So what is happening after 23rd day? So does the pain returns or it's just the variation?
Well, excuse me, I couldn't catch the some part of your question. Would you extend your question because I couldn't catch well?
I think somebody is asking about the graph where you plotted day versus pain. So he is asking whether after the 23rd day there is change in the perception of pain. Does it returns or it subsides? Something like that.
Sorry. I'm very sorry. I couldn't catch the question. I couldn't catch some important words.
Okay.
Would you type
another question? Yeah.
Okay.
It's in the chat box actually.
Would you type on the chat?
Yeah, it's there in the chat box actually.
Okay.
And there is another question. Meanwhile, we can take that. Is there role of dorsolateral prefrontal cortex in control of pain?
Yeah, there are some papers showing that dorsolateral prefrontal cortex is activated in some kind of human patients, not mostly in the patients with some phantom limb syndrome and so on. But I don't think that dorsolateral prefrontal cortex plays primary role in that in most of the chronic pain patients. Okay, thank you very much. So after that, if some superioritas. Yes, we don't know. Thank you very much. This is a very, very good and important question that we are trying to ask, because what I've shown you is a sensitization to touch. So this is tactile allodynia. So we have confirmed that tactile allodynia lasts about 2 weeks or more. And but we haven't done any behavioral analysis, such as like escape behavior or avoidance behavior, or we haven't measured the change in appetite, because chronic pain is quite that interrupts the appetite system, or some sexual activities, everything. So the honest answer to this question is we have not yet analyzed, but we are trying to make a system to analyze these other behavioral changes. We have just analyzed desensitization. So this is the honest answer. I hope this is clear to you, and I hope you understand there are a lot of things to do from now.
One more question. What is the evolutionary significance of this nociplastic pain?
It is quite important because, for example, it might depend on the definition of nociplastic pain, because in the for example lower invertebrates like octopus or like squids, okay? So there are papers, for example, if you cut one leg or one arm of an octopus, and they have some remote sensitization. So they show that widespread pain. You can imagine that. If you cut one of the octopus arm, they have some sensitization in other arms. So they have widespread sensitization. And this sensitization lasts some days, a few days. And there's a paper showing using a squid in that case. And if you cut one of the legs of the squid, and they show sensitization in the other body parts, okay? And they tried to fight with the predators, okay? So it's a strong fish that loves to eat the squids. So they put the squids and these predator fish in one water bath. And what they have observed was, squids try to escape from this predator, okay? You can imagine that. And what they did was they administered some analgesics to reduce that pain sensitization in this squid. So they injected analgesics to these squids, and the mortality of these squids, in other words, the probability that squids are eaten by the predators are increased by administering analgesics. So this means that this kind of sensitization or some kind of pain, something like that in the squids, help them to more sensitive or vigilant to escape from the predators. So this is one of the evolutionary reasoning that this kind of nociceptive plasticity or nociplastic changes, or in other words, some nociception-induced widespread pain would be important, would be beneficial to increase the survival rate. Does it make sense? I hope.
Thank you so much, Professor Kato. Thanks once again, and we hope to hear you offline someday in India in near future.
Yes.
Thanks a lot for sparing your time.
Thank you very much. Thank you. Dhanyabad.
I think Dr.
Thank you.
Thank you very much.
There is one more question by Dr. Abdul.
Okay.
There are more questions, I think.
No, just I want to say Dhanyabad, sir.
Namaste.
Yeah.
Namaste.
Very nice presentation. Thank you.
Thank you very much. Okay. Seems you're right. Okay. How are you?
Yeah. Everything good, sir. Going well.
So I wish all of you the healthy and productive days after this COVID-19. Okay. Thank you very much.
Especially the beginning is so good, we'll have that definitely.
Thank you. Dhanyabad.
So much. We'll now take a short tea break. I request all participants to rejoin by 11 a.m. for Professor Faguni Alvadi's presentation. Thank you. See you at 11 a.m. sharp. Presentation is by Professor Faguni Alwadi, who is a scientist at the Department of Clinical Psychopharmacology and Neurotoxicology at National Institute of Mental Health and Neurosciences, Bengaluru, Karnataka. She is the recipient of several prestigious awards, including ICMR Professor Surendra Mohan Marwa Award and the Jyotsna Moyi Raghunath Bhattacharya Prize. Professor Alwadi will speak to us on biomarker investigations in Parkinson's disease with cognitive impairment. Professor Alwadi's research includes neurodegenerative diseases like Parkinson's disease. Her research on biomarker enable early diagnosis of these diseases and also provide indicators of cognitive progression of patients suffering from Parkinson's disease. Welcome, Professor Alwadi. We look forward to an interesting session with you. Professor Alwadi, can you hear us? It seems Professor Alwadi is facing some technical issues. She will be joining in a minute or so. Request you to please bear with us with this technical glitch. Thank you.
Professor Faguni, you are the co-host now, so you can share your screen and you are able to talk.
Professor Aladi, your screen is visible, and I'm sure that you're audible too.
Okay.
Yes. Thank you.
So is that better? Yeah, I think.
Yes, yes, of course.
I'm sorry. Yeah,
Not at all, ma'am. Thank you.
Right. Right. So thank you for Professor Manjula, Dr. Geeta, and the scientific committee, as well as International Brain Research Organization for giving me this wonderful opportunity to share some of our findings, my team's findings in today's August meeting. So the title of my talk is Biomarker Investigations in Parkinson's Disease with Cognitive Impairment. I understand that there are a lot of graduate students who are attending this meeting. So for their benefit, we'll start with the classical definition of Parkinson's disease. So as it has been classically defined, it is a neurodegenerative syndrome with the hallmark features of motor impairment. Neurodegenerative means there is active loss or degeneration of the neurons that are involved in this particular aspect of motor movement. Right, so this includes, so the major motor symptoms of Parkinson's disease include bradykinesia, which means slowness of movement, tremor, which is more during the resting phase than the action phase. So if you give a cup of tea or something to an early Parkinson's disease patient, when he catches hold of the cup, his or her tremors come down. Then there is rigidity, which is stiffness, as well as postural instability. The diagnosis basically is based on the cardinal features of the clinical examination and a supportive history. So there are many evolving concepts about Parkinson's disease that many other features like restlessness or lack of sleep, anosmia, cognitive deficits, they generally precede motor symptoms. So before the patient develops any of the motor difficulties, the patient already has a prodromal period of sleeplessness or loss of smell, etc. And the presentation of Parkinson's disease is generally variable. So it differs from patient to patient and from population to population. There is a lot of variability in the presentation of diseases, and a few genetic aspects have also been identified. So now coming to the anatomy as well as neuropathology of Parkinson's disease. And this is a slice of brain, a coronal section of the forebrain. And these areas in brown are the ones which are affected in Parkinson's disease, including this. So this is the caudate nucleus. Together with putamen, they form the striatum. And this internal purple structures are the globus pallidus externa as well as the globus pallidus interna. In addition, the subthalamic nucleus is also involved in pathology, and the midbrain structure, substantia nigra, which is the source of dopamine, is majorly affected. So here you have two different aspects of substantia nigra. The substantia nigra pars compacta, which is a compact structure of a compact nucleus containing several neurons, as you can see over here, and the reticulator, which is the output nucleus of the basal ganglia. So put together, they are often addressed as basal ganglia. Now, what happens to these neurons, or what is the base of or source of pathology? So here, this is a midbrain section of a human, and here you can see this black structure, black band, which is the substantia nigra pars compacta. It is black because it contains a pigment which is called as neuromelanin, which is, as the name suggests, black in color, because of which the general black pigmentation or black band appears in the midbrain. This is under normal condition. Now, what happens is when these substantia nigra neurons, which we can see there's several of them in this normal pars compacta, are lost in the Parkinson's disease patients during the disease progression. As a result, this part of the midbrain achieves a pallor. That means they become pale, which can be identified by just visual observations. So in case of loss of these substantia nigra neurons, their innervation to the caudate as well as the putamen is reduced, which results in the loss of dopamine and causes the cardinal symptoms of Parkinson's disease. So these neurons also are known to harbor show the aggregation of a protein called alpha-synuclein, and it is also tagged with ubiquitin. So these are the hallmark features of Parkinson's disease. So if one were to define the Parkinson's disease as per the recent Parkinson's disease definition, it is characterized by the classical features of tremor, rigidity, and bradykinesia with impaired balance that I mentioned earlier. The onset is slow and the deficits are usually asymmetric. That means the tremors would begin in one part of the body. Say, suppose if it is the arms, it is generally either the right arm or the left arm. Very rarely you have bilateral involvement. There is inexorable progression and a sustained response to dopaminomimetics. Lewy bodies are usually found in the substantia nigra zona compacta, which I just showed, and neuronal loss predominates again in this particular region, particularly in the ventrolateral tier. I will explain the ventrolateral, the distribution of the various subgroups of substantia nigra in after a while. And this leads to decrease in dopamine in the striatum with a rostrocaudal gradient. So there is a loss across the rostrocaudal gradient. So say suppose this is the rostral aspect of the brain and this is the caudal aspect. So and this is the midbrain. So the maximum loss would be in the caudal aspect of the midbrain. So recently a task force has, or not really recently, maybe a couple of years ago, a task force has been constituted to update the definition of Parkinson's disease, and this is an ongoing process, right? So if one were to really compile the number of deficits or prodromal features or comorbidities that we see in Parkinson's disease patients, they have different sources and origins. So if you can see over here, in case of brain-associated comorbidities, there is anxiety, depression, cognitive deficits, dementia, apathy. As far as eyes are concerned, many patients have blurred vision, diplopia, olfaction, as I've already told you, is reduced. Then there is drooling, hypophonia, dysphagia. Cardiac involvement is also there. So is the case of stomach with reduced gastric emptying and constipation. Again, intestinal involvement is seen in case of with poor motility as well as constipation, bladder dysfunction, reduced libido. All these aspects being affected in Parkinson's disease patients, it is wise to say that Parkinson's disease is not just a movement disorder. So whenever there is something undesirable, say a disease condition, there are definitely fit to have some risk factors. So what is a risk factor? It's a factor exposure to which increases the probability of having or developing a disease. In case of Parkinson's disease, one can see that most of the Parkinsonian patients are aged, say they are above 60 years of age, hence aging is a major risk factor, and men are more likely to be affected in most part of the world, other than Japan, where women are more affected. Genetic inheritance has been described in 10 to 15% of cases. Middle-age obesity and sedentary lifestyle is a risk factor. So is ethnicity. Rural living, well water ingestion, and environmental toxin exposures are generally associated with pesticide exposure. So now this picture tells you that, there are certain numbers over here. So they are generally depicting the prevalence of Parkinson's disease in each of these populations. So this suggests that prevalence of Parkinson's disease has something to do with ethnicity. This is a completely different aspect of Parkinson's disease pathogenesis, which we have been, my group has been studying over the last 1.5 decades. And we have also developed an animal model, and this can be discussed on some other forum. Now coming to coming back to the pathology of Parkinson's disease, most of us know, and it is pertinent to know, that alpha-synuclein is the master manipulator molecule in terms of Parkinson's disease pathology. And this was ably depicted by Professor Heiko Braak and his team at the Goethe University, Germany. So what they did was they just took simple slices of brain, 100 micron thick slices of brain, and stained it immunohistochemically for alpha-synuclein molecule. And as you can see, all this brown color is the presence of alpha-synuclein protein. So they looked at different parts of the brain and even the enteric nervous system, and just basically quantified it objectively, and found that the maximum expression of alpha-synuclein was found in the dorsal motor nucleus of vagus. So this expression was seen even in the pre-symptomatic phase of the patients, and it went on to progress towards the higher brain areas. So in the stage 2, the locus coeruleus was affected, in substantia nigra was the third to come in. That means alpha-synuclein positive bodies were seen in substantia nigra at stage 3 of the pathological scoring of Parkinson's disease. Right. So how does how does alpha-synuclein aggregation take place? Now, there are several hypotheses which have been proposed about the highway to aggregation. One such interesting hypothesis is the formation of hairpins. So now this is the normal alpha-synuclein structure with the C terminus here, N terminus here, and around 38 to 53 point of alpha-synuclein monomers, a hairpin bend is formed, a hairpin is formed. Now, when two molecules with such hairpins come together, they go on to stick together, they become sticky, and then over a period of time, fibrils are formed. Right. So alpha-synuclein is not a non-useful molecule in nervous system, central nervous system. So under healthy conditions, it is involved majorly in the synaptic function. It is involved in endoplasmic reticular formations. It facilitates autophagic and lysosomal clearance. It also balances the oxidative molecules, and also is involved in mitochondrial functions. However, in Parkinson's disease, the presence of these monomers and polymers and sheets make a dramatic effect on the normal functioning of the cell, and all these functions which were normally facilitated by alpha-synuclein are then dramatically affected. So there is autophagic lysosomal disruption, there is oxidative stress, and much more aggregation of alpha-synuclein, and again causes synaptic dysfunction. Now, in last couple of decades, several therapeutic strategies have been developed to target many of these dysfunctions. Say autophagic and lysosomal clearance needs to be upregulated, aggregation should be downregulated, alpha-synuclein formation should be, monomer formation should be reduced, as well as the pinocytotic movement of alpha-synuclein between cells should also be reduced. So this has been achieved by several strategies, like for certain small molecules have been used, or intrabody nanoparticles have been used, or silencing RNA or antibodies to alpha-synuclein have been raised in order to stop the progression of alpha-synuclein induced pathology, or rather reduce the pathological progression. Now here I have listed some of those modalities, like reduction of extracellular alpha-synuclein, which has been achieved by passive immunization, but this was negative on the movement disorders scale, that is the UPDRS scale, which is used by clinicians to judge the motor deficits. The second modality was active immunization, where the active vaccines were provided to stimulate the B cells. But again, in the phase 2, this is in the phase 2 trial as of now, there was an attempt to block the misfolding of alpha-synuclein, yet we are yet to see the reports of that clinical trial. Blocking alpha-synuclein aggregation was attempted by nilotinib, but it was negative again, so it has been discontinued. There's been an attempt to reduce the alpha-synuclein synthesis using siRNAs and antisense nucleotides, but this particular study is just in preclinical phases, yet to reach the clinical phase. Similarly, there was an attempt to use monoclonal antibodies against alpha-synuclein for early Parkinson's disease, and this was known as the SPARK study, but unfortunately that has been negative again on the UPDRS scale and failed on secondary outcome measures, that is the safety and efficacy. So this trial failed when they assessed more than around 500 patients at the clinical stage. Now, as my title suggested, that we are talking about biomarkers for Parkinson's disease with cognitive deficits. And we recollect this particular figure, which I showed you earlier, where the several aspects of cognition are affected in Parkinson's disease over the course of disease pathogenesis, and that includes cognitive deficits. So this may include memory loss, or apathy, or lack of understanding by the patient, or loss of mathematical abilities, writing disabilities, and writing deficits, and all other aspects. So if we have to understand the cognitive impairment, we'll quickly try to see how does cognitive impairment fare in the Parkinson's disease patients. So it starts with just normal cognitive function, or no or minimal cognitive decline, and this is the early Parkinson's disease, and this probably it could progress into rapid progression, or mild delayed progression, or no cognitive decline at all. But in many cases, in case of Parkinson's disease with mild cognitive impairment, one can see this cognitive heterogeneity in terms of, let's say, a patient develops initially some cognitive deficits, then reverts to normal cognition, once again develops mild cognitive deficits, and then rapidly progresses to dementia. So this kind of heterogeneity in the progression of cognitive impairment has been seen in Parkinson's disease patients. And so what are the common neuropathologies associated with Parkinson's disease? So we all know that alpha-synuclein is known to aggregate within the dopaminergic neurons of the substantia nigra pars compacta. In case of Parkinson's disease with dementia, which also comes under the Parkinson's disease with cognitive impairment cohort, people have reported the aggregation of alpha-synuclein and Levy-like pathology, Levy-related pathology in the amygdala. There is a formation of cortical Levy bodies, that is presence of Levy bodies within the cortical neurons. And at a later stage, one can see striatal Levy pathology in the patients. 50% of the Parkinson's disease with dementia patients show presence of beta amyloid plaques, tau tangles, as well as presence of beta amyloid pathology within the blood vessels. So there is within the blood vessels, there is aggregation of beta amyloid, and hence this is known as amyloid angiopathy. So we all know that there are different transmitters, because all these different aspects of the patient of the brain function are affected, and hence there are different neurotransmitters that are associated with this cognitive decline. 3 of them are dopamine, noradrenaline, as well as acetylcholine. Sorry. So if you look at the dopaminergic system in controls, one can see a normal functioning of the dopaminergic system. These are the different brain regions that are seen to be innervated by these by dopamine. These are the regions that are innervated by the that are fed by the locus coeruleus noradrenaline, as well as these are the cholinergic regions. So in case of Parkinson's disease, now you can see the scale over here. So intense red means higher effect. So in normal Parkinson's disease patient where there is no cognitive deficit, one can see the darkening of these basal ganglia regions, and to a certain extent, the cholinergic system is also affected. In mild cognitive deficits, some amount of basal forebrain is affected, as well as very little effects on the locus coeruleus as well as the acetylcholine. Whereas in PDD, where there is intense dementia, majority of the regions are affected. Whereas in DLB, that is the dementia with Lewy bodies, dementia is the first aspect that is developed. So here you see hardly any motor involvement, whereas there is intense Lewy body involvement and cell loss as well as denervation in the cortical areas. Right, so why this concern about cognitive deficits in Parkinson's disease? We have once said that that Parkinson's disease is not just a movement disorder. So this is a summary of the cohort studies on prevalence and cumulative prevalence of cognitive impairment in the Parkinson's disease patients. So here, if one would see the PPMI, that is the most recent study, as well as the Sydney multi-center study, they have shown around 60 to 70% of patients develop cognitive deficits over a period of time. Many other studies have also reported cognitive deficits, in which ranges from anywhere between 17 to 43% at baseline. So that means that cognitive impairment is indeed an important aspect of the disease pathogenesis, and it needs to be targeted. So when you need to understand the pathological progression, or as a life science scientist, one is always apprehended with this kind of a question, that can we really do something? So there are several studies which have been focused on biomarkers for Parkinson's disease in an attempt to understand whether at any juncture of the Parkinson's disease pathogenesis progression, can we block or reduce the progression, right? So say there are certain parameters like sensory and olfactory testing which has been done, but it is it has an overlap between Parkinson's disease, dementia with Lewy bodies, and also in Alzheimer's disease. So that means it is not specific for PDD. Electrophysiological studies have shown some observations, but most of them have been based on small number of patients. Imaging studies are available, but they're expensive, very limited availability. Structural MRI also shows some kind of overlap of regional atrophy patterns between PDD and normal PD, whereas and as well as in dementia with Lewy bodies. So several other modalities have also been used. Genetic screening has been used, but I think till date the best result or the best outcome has been facilitated by proteomics-based studies, as well as other biofluid-based studies, like certain targeted proteomics have been studied, but and because they are evidence-based, they can be handled at a single patient level, and they're broadly available. They are also good research tools because we will understand why they are good research tools over the next few slides when we understand the pathogenic patterns of and pathogenic potential of the identified proteins. Right. So as we see over here, biomarker studies using biofluids of the Parkinson's disease patients have still been useful, but in many cases, the results have been inconsistent with no direct assessments on Parkinson's disease with dementia. So this is a summary of these targeted biomarker studies. So these, as we know, amyloid beta 42, total tau, and phosphorylated tau are generally associated with Alzheimer's disease, which we know is a disease majorly about cognitive deficits. So some studies have shown a reduction in their levels in the CSF. Some of them have shown an increase. Again, you see a heterogenic pattern over here. Now, alpha-synuclein, which we have been discussing thus far, is a putative marker for Parkinson's disease. Again, these studies have shown a mixed outcome. So, and also in the Indian scenario, we do not have sufficient studies to talk about their applicability as biomarkers. So other than targeted biomarkers, there are some non-targeted biomarker studies as well. So what are non-targeted biomarkers? So these are unbiased studies where proteomic analysis is done irrespective of a target. So we are not here trying to identify what happens to alpha-synuclein or what happens to amyloid beta. We're just seeing in an unbiased manner which of the proteins that is present in the CSF is either upregulated or downregulated. Now, this particular study, which used autopsied prefrontal cortex, showed dysfunctions in synaptic proteins. This particular study on CSF showed apolipoproteins being affected, and another study showed SERPIN-A1. Here again, we do not have a consensus on the outcome of the synaptic proteins. So now a quick recall. Right. So of all that we have discussed until now, we know that mild cognitive impairment and dementia are important aspects of Parkinson's disease pathology, and they tend to increase with age in years of the Parkinson of the disease progression. And 30 to 40% of PD patients develop mild cognitive impairment, where 2 or more cognitive domains are affected, but daily life activities are preserved. The more critical aspect is in case of Parkinson's disease with dementia, where the daily life activities are highly impaired. So, and that there is inconsistency in the knowledge on biomarkers for diagnosis of PDCI at an early stage. So now in the next few slides, I'll be talking about some of the findings of our laboratory on biomarkers. So let's see what is the definition of biomarkers. So biomarker is a defined characteristic that is measured as an indicator of normal biological process. That means this particular protein or molecule should be involved in the normal biological process within the organ of interest. Its over or under expression should cause the pathogenic processes, and it should also be a measure to estimate the responses to an exposure or an intervention. So if there is a treatment modality present, say something which can be given as siRNAs, then that particular protein expression should come down. Or if antibodies are given, again, the expression should come down. So this kind of a response to an exposure or intervention should be measurable. Right. So this particular slide shows the schema of our studies that we conducted. We initially, so the neurologists at NIMHANS Movement Disorders Clinic, they recruited the patients using several criteria, that is the UK Brain Bank criteria for diagnosing Parkinson's disease patients. And we also included some normal pressure hydrocephalus patients as internal disease controls, because many times these patients also present with some motor deficits. Now, all these patients underwent neuroradiological screening for particular for the features that are generally seen in these patients, and thereafter the CSF of these patients was collected by lumbar puncture. And here again, we introduced one more group, that is the non-neurological controls. These are patients that were undergoing plastic surgery, but had to undergo spinal anesthesia. So lumbar puncture was possible for them to collect the CSF. And these, as I mentioned, were collected at NIMHANS. Right. So these patients, after being recruited, either they were controls or they were the diseased patients, they were assessed for their neuropsychological performances using MOCA, Montreal Cognitive Assessment Scale, with a scale of less than 26 was considered as a cutoff for cognitive deficits. And then detailed analysis was performed using neuropsychological battery. We then subjected these CSF samples to QTOF LC-MS analysis, got the identification of proteins and statistical analysis. Thereafter, the proteins which were maximally affected were then validated by ELISA in the same CSF samples, and we chose 2 proteins out of these 5 specific proteins to test their pathogenic potential using a mouse model. So the first aspect, the demographic details of these patients. Here we can see the mean age is fairly comparable between the control PD, PDCI, and NPH. This is the mean disease duration in case of the Parkinson's disease and the NPH patients. These are the MOCA scores. So you can see the PDCI and the NPH show deficit in their cognitive abilities, and these are the UPDRS on and off scores. So these show the representative MRI. Here, this is that of a PDCI, and you see cerebral atrophy, which is proportionate to age. And this is from NPH. So this is normal pressure hydrocephalus. So you can see the ventricular enlargement. You see this white space is a ventricular enlargement, and it is full of CSF, whereas the ventricles are fairly small over here, which could be age-related ventricular atrophy as well. So the tests that were conducted were pertaining to attention, language, memory, executive function, visuospatial function, and these are the details of these tests which were conducted, and these were conducted under the observation of a clinically trained clinical psychologist, right? So when we looked at the cognitive functions of compared all these patients for these different executive functions, memory, language, attention, and visuospatial function. So one can see that in the PDCI as well as in NPH, there was a major dysfunction in several aspects. Barring the language, I guess, all the 4 other aspects were affected. Now coming to the proteomic study. So all the samples, as I mentioned earlier, were subjected to proteomic analysis. So this is the heat map of these CSF samples collected. These are the names of the samples, rather the codes of the samples which were analyzed. And this is the heat map. So what we found was that out of the 281 proteins that were identified, around 42 were differentially altered. So these are the volcano plots, and one can see over here on the right are the proteins which are upregulated in these particular diseases. These are tabulated over here. So this Venn diagram shows that 7 proteins overlap between these, but there were 9 proteins which are listed over here, which were very specific to Parkinson's disease with cognitive impairment. That included the fibrinogen alpha chain, apolipoprotein A1, and those that were common were fibrinogen gamma chain and beta chain. So, the other proteins were gelsolin, complement factor H, and apolipoprotein A4. So what we did is we selected these few proteins, that is the fibrinogen, gelsolin, complement factor H, as well as apolipoprotein A1 and A4. So as we know, fibrinogen is a coagulation factor, so that is, it is involved in normal functioning as well. Gelsolin is also known to coexist with alpha-synuclein in the Levy bodies. So that means it has some role in disease pathogenesis. CFH is a complement regulator. It was found to be more specifically upregulated in PDCI compared to NPH when we looked at the mass spec analysis, and APO-A1 and APO-A4 were dysregulated in PDCI, and they are otherwise involved with cholesterol transport. So now looking at the CSF ELISA. So I'm sure all the graduates as well as postgraduates know that ELISA is a modality to estimate the protein concentration within a particular biofluid. So here we can see that fibrinogen was much upregulated, at much higher levels in PDCI as well as NPH. Now in PDCI, it was not statistically significant. However, it still showed a 6-fold increase, right? So that means this was important. That could have been because here we have used a fibrinogen whole molecule, and the upregulation that we saw in mass spec was specifically for fibrinogen alpha chain. Coming to CFH, our observations were validated. We found much higher higher levels in the PDCI cohort, and in gelsolin it was reduced compared to PD. So that means it can be used as a discriminatory factor to distinguish between PD and PDCI. APO-A1 showed an upregulation in NPH. However, the ratio was much better appreciated. Like here one can see that when you compare PDCI with NPH, the ratio was much higher in NPH. So that means this again could be used as a discrimination factor between PD to distinguish between PD and NPH. So now what emerges is that there is no single molecule that has been that can be vouched upon to be a biomarker for Parkinson's disease with cognitive impairment, right? So there has to be probably a combination of 2 or more molecules. So we also performed a correlation analysis between the protein concentration, that is the fibrinogen concentration, and the Wisconsin test percentile. And here we can see that the higher levels of fibrinogen are associated with lower function on this scale. So a quick understanding of the results so far. So we found that fibrinogen, CFAH, gelsolin, and APO-A proteins were significantly altered in PDCI. CFAH could help distinguish between PDCI and PD. And the APO-A1 ratio was more implicated in NPH. Fibrinogen levels in the CSF thus may help predict the cognitive decline in the executive domain. However, because probably the cohort size was small, we did not actually see a correlation between the levels of any of these proteins and the disease duration or the UPDRS scales of the patients. So now, coming to the important aspect of the biomarker. So if we use any molecule or a protein as a biomarker, we intend to use it for biomarker, to categorize it as a biomarker. It should have the ability to explain the steps involved in disease pathogenesis. That is, when injected extraneously, it should have the potential to induce disease-like symptoms in the animal model. So this brought us to our next modality of study, that is assessing the behavior of the animals. So now this is these are the videos of the pole test. Now on this pole test, one can see this is a vehicle injected animal. So this is a pole test. So this is just a vertical pole of 50 centimeter height, and the animal is allowed to walk down. So as we know, the animal has to use its grip to come down. So that shows its ability to traverse on the on the pole. And now here we can see this is the fibrinogen injected mouse, which is allowed to move. And this is a CFH injected mouse, which is. So if you look at this mouse, which is coming down quickly, this mouse takes a little longer time. And let's see what happens next. We go to the pointer options. There's a pointer. How do I go out and yeah, so these are the histograms that show the time taken by the mice to come down. And as you can see over here, we use 3 different concentrations of fibrinogen. We began with fibrinogen and we found that this was not adequate to induce those movement deficits. And here, the fibrinogen 2.5 microgram as well as fibrinogen 5 microgram animals took a little longer to come down the pole, which was not the case after CFH injection. So studies thus far have been published. This is a recent article which is available online. So now, as we saw, CFH injected animals were not so deficient on the pole test. So they were able to come down. So they probably had a good grip and could walk down. We wanted to do further analysis of the gait. Gait is and the ability of the animal to move on a particular platform in a particular direction for a particular distance. So here, as we can see in this in this particular well, so will not waste time on you would have seen at the initial aspect, I think, that the mouse was moving. So the mouse moves on this particular platform, and the right foot, the right hind foot, the right front foot, the right hind, the left front, and the left are demarcated here in colors, and these are the movements. So if you can see the control, there is a particular pattern of movement, whereas in fibrinogen injected animals, there is kind of a drag, and which is much more visible in the CFH injected animals. So this is how the animal moves, and this is that is tabulated. Yeah, so these are the analysis which have been done on different feet of the, paws of the mouse. Here you can see there was a significant decrease in the stride length of the CFH injected animals, and so is the case with fibrinogen 2.5 injected animals, which is not very much evident in the other limbs, other than the left front paw, which is again affected in the CFH injected animals. So this suggests shuffling of gait, which is a prominent gait deficit in the Parkinson's disease patients. And it is probably, as you can see here, it is in the RF and the LF. So that means in the forebody of these mice. Now, this suggested that these two proteins are able to induce pathogenic features in the mice upon injection. So now in Parkinson's disease, with cognitive deficits, we know that cognitive impairment is an important trait. So we need to see whether, in addition to the motor deficits, whether these particular proteins have the capacity to induce cognitive deficits as well. So the novel object recognition test is a very good parameter to understand these cognitive deficits. So we performed the cognitive So we performed this novel object recognition test, and as you can see over here, this is how it is put. So initially, two similar types of objects are placed, and the mouse is allowed to explore this particular arena. And after initial habituation, we replace the familiar object. So here in this case was a green colored cube was replaced with a triangular object, which is red in color, and the animal was allowed to explore. And what one could see was that in case of fibrinogen injected mice, there was a reduction in the differentiation index. So it did not spend sufficient time with the new object. Same was the case with CFAH injected mice. So now if one would estimate the percentage of exploration, because here there are two aspects to this test. One is the mobility as well as the exploration. So we wanted to see if there is any effect on the mobility or effect on the exploration. And here one could see that in the CFH injected mice, there was a significant reduction in the overall mobility, which suggests that these molecules were able to induce some amount of cognitive decline or probably lack of motivation as well as motor deficits. So to summarize the behavioral analysis on pole test, we saw the movement was impaired, which could suggest bradykinesia or postural instability. Gait analysis, analyzing the stride length, showed shuffling of gait, and NOR analysis showed cognitive decline and loss of motivation. So fibrinogen again here was able to induce impairment in all three parameters, whereas CFH induced impairment in 2 out of 3 parameters. So now this was the proof that these animals are affected. They are not, they are not competent enough, like the control animals, to perform certain tasks. So the next was, we had to see whether there was a pathological loss of these particular areas or of the basal ganglia, as well as the cognitive of the areas that are involved in cognitive functions in the mice brain. So here we counted the number of dopaminergic neurons. Now this is the substantia nigra pars compacta. This is the VTA in different injected animals. And here we can see that fibrinogen 2.5 induces a loss of dopaminergic neurons. And whereas in CFAH 2.5 injected animals, there were quite a few number of degenerating neurons. So these are the degenerating neurons. And then we looked at the morphology of the neurons, and we found that there were both degenerating as well as hypertrophied neurons in the fibrinogen injected animals as well as in CFH injected animals. Now, we also know that different regions or subregions of the substantia nigra project to different regions within the striatum. So this is color coded over here. So the green goes to, so the VTA and the medial substantia nigra, they project to the medial striatum. The dorsal aspect supplies to the to this particular band, and the lateral substantia nigra supplies to the dorsolateral striatum. So these are the different subregions of substantia nigra. Here's the substantia nigra pars reticulata. This is the compacta. This is the PBB, and this is the VTA. So what we found was that fibrinogen caused a neuronal atrophy in the dopaminergic neurons of the lateral aspect of substantia nigra pars compacta. Similar results were seen in the dorsal aspect of substantia nigra pars compacta. as well as in the medial aspect. So now in the medial aspect, what has happened is that the expression of the TH has also come down. That means has been reduced. That means there is a loss of dopaminergic phenotype. That means it has its potential to synthesize dopamine has gone down. Now, the VTA that is associated with the cognitive and the reward centers of the brain, there we saw there was an increase in the size of these neurons, so which could be a compensatory hypertrophy. That means when in presence of an insult, the neurons try to increase their potential performance. These are the changes in the striatum. Here we can see there is a loss of dopaminergic innervation. There's a reduced intensity of TH expression in the fibrinogen injected animals, which is seen in the histograms over here. So the next, we wanted to see the neuropathological correlates of cognitive deficits. Here in this case, because the dopaminergic innervation is seen maximally in the CA1 and the subiculum fields of the hippocampal formation, we looked at these aspects, and the volume of this of the CA1 was fairly preserved, but the morphology of the nucleus and the cytoplasm was affected. As you can see, there is a nuclear hypertrophy, and there was chromatolysis within these cells. They were these are degenerative and necrotic cells that can be seen in these cells that are from the animals injected with these neurotoxins. So the subiculum was fairly well preserved in terms of volume, so were the lateral and the third ventricles, suggesting that this is more of a PDCI-like pathology, but not NPH-like pathology. So coming to the conclusions, the number of neurons were reduced in the substantia nigra pars compacta. There was a loss of dopaminergic phenotype. There were hypertrophic dopaminergic neurons, which could compensate for the dying and dysfunctional neurons, and there was a loss of dopaminergic input to the striatum. And there were hypertrophic neurons again in the CA1 and subiculum, which could be there to compensate for the gradual cognitive decline. So these are the hallmarks of Parkinson's disease, and the hippocampal formation showed the deficits which are hallmark of the cognitive impairment. So collectively, fibrinogen was able to induce impairment in almost all the 5 neuropathological aspects that we studied, and CFH was able to bring about impairment in 4 out of 5 aspects. So this is the final slide to summarize our complete study. Here, through the proteomic analysis, we found that fibrinogen and complementary factor H were upregulated in the PDCI CSF. And when these proteins were injected into the mice failed to perform satisfactorily on pole test, and their strides were affected, so were their cognitive parameters. And there were other neuropathological parameters like the impact on the substantia nigra pars compacta, striatum, as well as the hippocampal subfield. So amongst the two, although it might be a little premature to say, fibrinogen was still much more impactful than CFH. And as we saw, fibrinogen levels were also much higher in the patient CSF that we had analyzed. And hence this could be 2 good biomarkers to understand Parkinson's disease with cognitive impairment, and even for future studies on them. So while I thank you for your patient hearing, I'd like to acknowledge the team which was involved in this entire study. Firstly, the funding agency, ICMR. So we had a grant from ICMR, and Aditi, who was the student who conducted most part of it. I mean, so she conducted all the MOCA analysis onwards. As we know, the neurological deficits were earmarked by the neurologists, Psychological deficits were quantified by the clinical psychologist, and the rest of the study was basically conducted by Aditi. We had biostatistics support for unbiased analysis of all our observations. Imaging was performed by Professor Jitendra Sen. Now, we collected, as I mentioned, CSF from control patients, non-neurological controls from the Department of Anesthesia, Bangalore Medical College and Research Institute, and the proteomic study was conducted under the guidance of Professor Utpal Tatu at the Department of Biochemistry, Indian Institute of Science. So we also thank some summer trainees who dropped in to learn these techniques and perform some analysis. Thanks once again for your patience here.
Thank you, Dr. Falguni Alwadi, for a very interesting and very important topic for today. There are a few questions. I'd like to read them. Dr. Ansari wants to ask how alpha-synucleins get misfolded. So like from normal physiology to the pathogenesis. And
Yes.
is there any evidence or study for it?
Yeah, so there are several hypotheses which say that certain ends of the, C-terminal or the N-terminal ends get sticky, and hence they get misfolded. Actually, it's $1 million question which is yet being investigated. And as I showed in one of the slides, there is a hairpin bend which occurs at the, I'll just show you quickly. Yeah, so here, as you can notice, so region 38 to 53 of the alpha-synuclein monomers. So monomers are present and they may not be as pathogenic as the beta sheets that are present within the cytoplasm and even in the neurogenic, within the neuropil. So these kind of hairpin structures are formed, which then becomes sticky. So hairpin structure of one monomer comes closer to that of another monomer and they fuse together. So these kind of continuous fusion of several molecules causes the formations of fibrils. So here this is at the aggregate stage and this is at the fibril stage. And thereafter it's very difficult to dislodge them and or even clear them through autophagy. So that affects that affects the whole process. I hope that answers his question.
So there's another question. They're asking whether the motor disorder affects memory. For example, less interaction time of the treated mice with novel object could be as a result of motor impairment.
Yes. Indeed. So as I mentioned over here, we looked at the total exploration. Yeah. So here we have analyzed the total exploration time. So this particular CFH group, CFH5 injected mice, as well as the fibrinogen 5 injected mice, they show much lesser exploration. So they're not moving. So if they're not moving, it is likely that, it could be because of a motor deficit. So that's precisely what we have also mentioned, that it could be cognitive decline, it could be motor deficit. Now, this aspect of motor deficit is affected. There is shuffling of gait, but they were able to perform fairly, at a competent level on the pole test. So indeed, it is possible that their movement is reduced because of motor deficit. Thank you. I hope this answers the question.
Are these biomarkers nowadays used in clinical practice?
I hope so. I hope they soon enter into clinical practice because I I'm yet to come across studies which have provided convincing evidence, as in our case, about their involvement in pathology. But several clinicians have expressed interest.
And can they be?
Yes.
Yeah. Can they be used for target?
I'm sorry, I'm losing your audio.
I'm sorry. Can they be used for targeted therapy?
Yes. So the next question that we have asked is, will be, so it's, we are going to try a patented molecule. So for the sake of, for the benefit of this, the future study, I won't be able to disclose the name, but we are going to target processes where fibrinogen and CFH are involved. So that at a preclinical stage, we can check whether these molecules indeed, if these molecules are targeted, whether it can be, extend a therapeutic benefit. Yeah, thank you.
Another thing I wanted to know, have you developed a biomarker panel kind of a thing with multiple biomarkers in it to diagnose cognitive deficit?
These are questions which are going on with the industry. So we have discussed these points with some of the people who make arrays as well as ELISA plates, so that we can have multiple markers being targeted estimated at the same time.
Thank you. Thank you so much, Dr. Fandana.
My pleasure.
It was a wonderful presentation.
Thank you. Thanks for providing me with this opportunity.
Thank you, Professor Alardi. It was a wonderful conversation. So our next speaker is Professor Kocher, who is a professor and faculty in charge of Neurocognitive Lab, Department of Physiology at All India Institute of Medical Sciences, New Delhi. She's also spearheading the yoga intervention protocols at the integrated health clinics at AIIMS. She'll speak on the topic transition in nutrition and brain-gut axis, neurobiology and psychobiological approaches. The gut microbiota bidirectionally impact the central and enteric nervous systems, and alterations in the gut microbiota may impact the brains of patients. Professor Kocher's presentation will try to fill this crucial gap in gut-brain axis. Professor Kocher, it's a pleasure to have you with us today. The floor is all yours.
Hello, good morning. Can you hear me?
Yes, ma'am, you're audible, but not visible.
Yeah.
Right. Can you see me now? Yeah.
Yes, ma'am, you're audible as well as visible. Thank you.
Thank you, Imran. So first of all, it's an honor, and I thank Dr. Geeta Trilokumar. In the morning, I was seeing her presentation as part of the DBT Alliance on womb health. And Dr. Manjula is a very dear friend. She did her PhD in our department, and all the Dr. Ansari is here, and I really congratulate and thank IBRO for this, another IBRO-APRC specifically, and I think this is my third or fourth presentation through these IBRO schools and workshops in these past months. So I also thank Dr. Faguni, and I told all my world-class lecture presentation she made, and I quickly edited some of my, changed the topic because originally it was posted as brain gut axis in Parkinson's disease, but now, as has been rightly said by the previous speaker, it is the enteric nervous system, and therefore I wanted to see the different dimensions of food, gut, and brain. Next slide, please.
Connectivity problem.
Yeah, though it hasn't been mentioned in the title, Sharma and Ms. Sneha are also authors of this presentation. So, neurodevelopmental, neurodegenerative and psychiatric disorders and their therapeutic interventions both have distinct signatures on the brain-gut axis, and understanding these interactions has brought new insight both in the evolution and development of the disease, the progress of the disease, as well as we all know that many times when we say that there was no result or not much result, I just want to tell all the dear students at IBRO that these days the scientific community believes that negative finding is equally important, if not more, because that way we don't spend too much time and resources and effort in doing, and we change our directions. And some of my slides towards the end will also tell us how we do it. So when we say traditional diet, like in psychology, we have culture-based psychotherapy. Similarly, we feel that taking from our traditional resources, the microbiome is influenced over generations, okay, and epigenetic modifications. Immigrants who migrate to other countries, their microbiome changes according to the geography, and this also has a great role in non-communicable diseases and also neurological disorders. We wanted to see that whether traditional Indian, because I worked in nutrition, my PhD is on Indian dietary spices. So the three main components of Indian traditional diet, one is the coarse cereals, second, the use of dietary spices, and third, use of fermented foods or what we say probiotics, whether in the North Indian population or in the South Indian, East or West. So we wanted to study these things in the scientific way, and as is said here in the last line, probiotic interventions induce epigenetic modifications. Dear students, you all know that now the dogma of DNA being the master has been broken, and as Dr. Fagni also referred about silencing RNAs, so we know that the expression or de-repression, okay, or induced repression of certain DNA can be done, and that is one step further in our nature to nurture debate. So neuroplasticity, there disorders, learning disability, autism spectrum disorders, ADHD, as well as a host of psychological disorders, including psychiatric morbidities. Next slide, please. Yeah. So I will just quickly go through this, that the food we feed a child from in utero has a significant impact, and as my previous speaker very nicely explained the biochemistry, also the basics of nutrition. We give seed or we give nurture to soil, but then we need to know what are the components of the soil. How many minerals are there? What was grown before that? So what are the diet options? And now one word you must remember, students, that is nootropics. NOOTROPICS. Cognitive enhancing foods. Some are natural, some are synthetic or man-made, for nutrition boosting, both for physical and mental health, and therefore diet-based approaches are seen. Next, please. So I can quickly run through this because basically this is part of nutrition. And the only thing I wanted to say is that traditional diets had great wisdom. Yeah. So these help in learning, they help in assimilation, and they help in improving our protein content. And also I wanted to say that as both a nutritionist as well as a neurophysiologist, I would like to say that all fat is not bad and sunlight is not bad. Next, please. Yeah. So now coming to the scientific basis of brain-gut interaction or enteric nervous system. We don't like to spoon feed our students or delegates. So I want to give you a little bit of a homework. Go home and read about the 7 or 8 ways in which the vagus nerve contributes to a bidirectional brain-gut axis interactions. Okay? So many of the foods which we eat are traditional foods. No? Like one big example I can give you is from the Chinese cuisine, the sodium monoglutamate, ibotinate. Many of these flavor enhancers are also acting as neurotransmitters, and gut from the metazoan evolution has been part of neurosensor. Okay? Yeah. Next slide, please. So epigenetic can cause methylation or sulfation and like that. So let us now just study a few more goalposts in the gut-brain partnership before I tell you what we do in our laboratory and invite each one of you to visit us, to communicate with us, to further the cause of this science. Next, please. Yeah. So, even in English language and literature, we know we have a gut feeling or an intuition or an intrinsic sixth sense. So, microbiota changes are one which can cause the epithelial breakdown, or what the barriers are in place, they break down, and then there is immune activation, okay, because the bacteria which are in the colon, okay, they are just like lipopolysaccharide stimulation is a test for testing immunogenicity. So endotoxemia can occur, which can lead to pro-inflammatory cytokine productions. And then the other barrier, which is the blood-brain barrier, can break down and alter the neurotransmitter levels and cause changes in synaptic plasticity, and then lead to neural effects, behavioral as well as psychological or mood changes. And these are bidirectional. So stress or dietary or environmental triggers can affect this bidirectionally. So it is up to us whether we want this to be a vicious cycle or a virtuous cycle. Next, please. So the GI, or starting from the oral mucosa or the buccal cavity, yeah, is the first physical barrier or defense against bacteria and viruses. And as my dear young lady Shilpa Sethi says, probiotic ki ad mein she says, oh, maximum immunity is in the gut, But because of COVID, the word immunity has been almost used very, very loosely, not in the way scientists like. And I want you to take all this with a pinch of salt. Okay? In science, we can't just feed somebody immunity. Everything which is marketed is saying immunity booster, immunity khila rahe hain. So we don't khilao immunity. We khilao food, or we have some medicines, or we have some interventions in diet which can cause a lot of changes in our systems-based approach, which can lead to enhancement of either acquired immunity or even innate immunity, or whether it is a B cell mediated immunity or a T cell mediated immunity. So the GIT is the first exposure to the external environment, at least in humans or mammals, and all nutrients are precursors for neurotransmitters. Right? And we all know about tryptophan, we know about serotonin, and we know serotonin, we know dopamine. adrenaline, acetylcholine. All of these 5 histamine neurotransmitters are what we call diffuse neuromodulatory systems of the brain. These are the ones which diet first affects. Okay. We also have projection neurotransmitters in long pathways like GABA, like glutamate, and all of you know from your biochemistry in school about essential amino acids and why we call them essential. Next, please. So, a healthy brain and a healthy gut are a bidirectional, complementary, combinatorial relationship, and stress can affect satiety, alterations in behavior. Sometimes people eat comfort food. If one A gets stressed, he or she may eat more food. If B gets stressed, they may actually be off food altogether. So, in biology, we usually don't follow linear equations, we follow non-linear systems where emergent properties come. Many times things are bipolar. And I remember Dr. Manjula Suri's thesis, when she was working on sugar and analgesia in our department. And many times, young children were given before injection, the mother would give them a toffee or a drop of honey or some jaggery, and how and the beautiful thesis she made where she showed how endogenous pain modulation pathways and how sugar can have a bipolar kind of an effect, both analgesia and hyperalgesia. So I wanted to say that when we consider diet, many times the effect can be bipolar. So don't try to extrapolate linear. There are some doses which are physiological, some which are supraphysiological, and some which are pharmacological. And then there are effects like tachyphylaxis. There are other effects of tolerance also. So it is not a very simplistic explanation. So gut peptides and microbiota can all affect the secretions in the GIT, the movements in the GIT, the blood flow, the sensitivity of the receptors, homeostasis. And one thing I wanted to tell all my young participants today is TRP channels, the transient receptor potential channels. In the early 90s, when I was doing my PhD, these things started to come about vanilloid receptors. And vanilloid also is part of what we call herbs and spices, and how these multimodal receptors, that is stimulated both by mechanical stretch, also by changes in temperature, also by the chemical component. So these are some of the ways in which the traditional wellness wisdom is now being evidenced in a platter as a scientific evidence-based medicine. Next, please. Yeah, so these are some of the pathways, whether it is by the hypothalamic, hypophyseal, pituitary axis, by inflammation, by dysbiosis, how mood is affected, how cognition is affected. Next slide, please. Yeah, so traditionally one said, oh, this disease is incurable. Okay, this is a degenerative disease, or this is a neurodevelopmental, and the whole life the child will have to bear with it. But now we know, we are understanding, like Dr. Fagani very nicely explained, how we develop and study, and then we do the theranostics, and then we do the therapeutic intervention as potential or putative helpful agents. So now the modern view is that things are modifiable. There is a plasticity, just like we have neuroplasticity. We have immunoplasticity, we have a diet plasticity, we have a Next slide, please. Yeah, so yeah, we can go to the next one. So genetic and environment, nature and nurture, this can occur at one end, that is the child or neurodevelopmental. This can also occur at the other end, that is neurodegenerative, and in a branch, that is a psychological, because the microbiome can also act as a psychobiome. Next, please. Yeah, right. So by paying attention to not only the learning part, but also to there's a branch. Previously we used to call it psychosomatic medicine. Now it is called consultation liaison psychiatry. So we can significantly improve the quality of life and level of functioning. Okay, and basically for all degenerative or developmental disorders, only these two things are needed, that quality of life is good and functional outcomes are improved. Next. Yeah, I will now quickly go from neurodevelopmental to actually what we do, so we can run the slides further. Yeah. Yeah, so we all know, and you can all read about gut mucosa, how dietary spices affect this, how coarse cereal diet affects this, and how the probiotics affect this, and also traditional phytomedicines affect this, and then how they affect the brain, and how the brain, whatever is affected in a systems based approach, it does a good or a win-win situation for our liver metabolism, for our kidney metabolism, okay, and other body metabolisms. Next, please. Yeah, so immune dysregulation, increased inflammation, okay, can occur, and this happens not only in the autistic thing, but as we know about, and very nicely explained by Faguni, alpha-synuclein, or there are so many illnesses which we now group under a term called alpha-synucleinopathies, and how, as I told you, the homework, see how vagus can affect in 7 or 8 ways this, so people can ask how there is an organic connection between brain and gut, okay, and how this becomes two-way, and how the hypothalamic hypophyseal axis also modulates this. Next, please. Yeah, next. Yeah. So let us now shift from the gut to the brain. So probiotics have many types of actions. This is an old slide, and now much more is known. Two PhD students are working, one in the human in a clinical trial, one on the animal model. And not only are we doing probiotic interventions in our laboratory with my colleague, who is also the advisor and mentor for this workshop, Professor Suman Jain, and a great colleague of Dr. Manjula Suri, we are also combining one stream probiotics with other stream of magnetic stimulation, and seeing that how these traditional approaches of using certain kind of metal vessels for food, okay, or wearing certain kind of magnetic material bracelets, how now we can quantify it, objectify it in controlled doses of controlled frequency and give transcranial magnetic stimulation. And the latest one is what we call theta burst stimulation. So my theme is to join the traditional practices and present them in the modern scientific context, and then see what result is there, and to show it to the Western world. Next, please. Yeah. Yeah. I have already told you how neurotransmitters are affected. We have, I think, about 5 or 10 more minutes, and I don't want to keep the next speaker waiting. So I can quickly now run through the slides. My job was, no pun intended, to give you a flavor of what happens in the gut. Next. Yeah. Next, please. I can share these slides with you on my email, through my email. Yeah. So also mental health. irritable bowel syndrome. Many mental disorders have GI manifestations, and many GI manifestations or GI disorders have mental manifestations. Both run hand in hand. Next. Right, right, right. Okay. Next, please. So similarly for one illness, which is schizophrenia, which is a psychosis, so also for bipolar, which is also a dementia, okay, along with it, neurotic disorders like anxiety, all are affected through the gut interactions, okay. Next, so also for aging, and neurodegenerative disorders are what we call accelerated aging, okay. Next. And we have traditional practices of using dahi, using fermented foods, using certain kind of chutneys and all, pickles and like that. Yes. So we all know antibiotics, then we know probiotics, then we know prebiotics which produce probiotics like dietary fiber, and then we have synbiotics, and also now we have psychobiotics. Next. Yeah. So this is very, very basic. You all know it. Next. So now I shall introduce you to some tools and techniques which we use in our laboratory. Next slide, please. Yeah, we'll just spend a minute on this. So various domains of cognition measurement. I feel cognition can be sensed by attention, which we do by visual tracking. We show images of food of various types and then see how long the person's eye attends to this food by reaction time, by duration bias, and by direction bias, because we put a same pixelated image of a non-food item on the screen. While we are doing visual tracking, we can also measure the brain mapping of the individual by FNIRS, okay? And we can also test the memory by giving certain tests. We can see which areas of the brain light up by functional near infrared spectroscopy. That means, and we are doing these studies for diabetes, then yoga intervention. We are doing these studies for caries in children. We are doing these studies for craving, for substance abuse. 2 or 3 studies we are doing, where we are showing the cues of a alcohol bottle or a substance abuse drug and see how the person, while the counseling and the behavioral therapy and the medicine psychiatric are going on, okay, to deal with these obsessions, we are providing tools and techniques to our clinician friends to evidence these benefits through the brain mapping. We can also, number 3, check executive function choice and decision making by giving a Stroop kind of task, or the subject has to press one of the 2 buttons. We also do the 4th domain of cognition apart from attention, memory and choice making, that is motor planning and performance, which we call motor cognition, which is the final common pathway by pioneering bare shaft cortical potentials or movement related cortical potentials. You can see some of my studies by googling my name, Kanwal, Kanwal Preet Kochar, and get to these studies. That is also your homework. And then we combine it with certain cognitive toolkits and questionnaires with the help of my psychology friends, like Faguni had showed you, the MOCA assessment, or the UPDRS score. Then we also do a wireless physiological monitoring of autonomic profile, respiratory rate, ECG, EMG, the electrodermal activity in the skin, the blood flow, skin temperature at 2 sites, to see what is the autonomic profile, and then we combine it with the body impedance analysis or body composition analysis, because we are using not very hi-fi instruments of the middle range, so that we can spare neuronavigation and actually we can customize according to the Indian morphotype or the anthropometry. So this is how we combine these and do various types of studies and help our clinician friends in seeing the effects of their intervention. Next. Yeah. So this is just the instruments and how we can see this. Next, we are doing about 25, 30 odd studies now. Yeah. So this is wireless monitoring. We have respiration and heart rate and physiological markers, and we combine it with the brain cortical activity. Next. Yeah. So these are some of the parameters which we measured, as I told you, for neuromodulation in obesity. Next. And so also for bear share potentials we are doing. Next. Visual tracking. Now we have an advanced equipment better than this, which stabilizes the head also. So epigenetics I already explained. Next, please. Yeah. So I had done a PhD in spices, and we had done a lot of work also. Some publications are also there of how the spices can act on various body systems, including brain and mind. Okay. Next, please. Yeah, one thing I wanted to say, which has, I think, not been shown here, that is in silico modeling. Many times, as it was shown by the previous speaker also, we do clinical trials and not much comes. So if we can do in silico modeling by running, say, what my student Sneha is doing, she's working on rat model of Parkinson's disease and seeing the phytomedicine. So if we do the pharmacogenomics or the phytogenomic databases, and we combine it with the protein databases, and then we can see in silico by running through the software, and then see that for the Indian genotype or phenotype and for the particular phytomedicine plant, which enzymes it actually is targeting, and then we devise our clinical trials based on our inputs or leads from in vitro or in silico modeling. That shall be the best situation. And thank you very much for patient hearing. You are always welcome to visit our lab, and you are always welcome also to join as summer trainees. I again thank IBRO, Dr. Geeta, Dr. Manjula Suri, and Imran for helping me and honoring me. Thank you.
Thank you, Dr. Kocher. It was a very interesting, very informative presentation. And there are a few questions. There's one question: will the overuse of antibiotics will affect the brain function? And will vegetarians who miss on few nutrients are affected in any way?
See, this is like, as you use the word, we cannot say by one each. It will depend on the person's, what you call family medicine, or what is personalized medicine, and in older systems we had constitution like Vatafa, Pitta. Now we have the person's microbiome, the person's basic nutrients. And one thing I also wanted to tell, maybe some part of the answer lies there, is that biomarkers, we have lot of statistical deviations and variances depending on the person's dietary history or the person's grandmother's dietary history and the person's local cultural history and populomics, as we say. So the cyclic regulations are more robust, and that is why when we measure and combine all of these using big data science help, that can give us the real answers. A very reductionist view cannot give us the real answer, can only give us some leads.
Thank you, ma'am. Another question I wanted to ask was, are there any side effects of pre or postbiotics? Because it's quite in use nowadays.
Yes, Manjula, you are very right. Just like we have a pharmacovigilance and we have a rational drug use and a rational antibiotic use, otherwise microbial resistance can occur. These interventions also have to be taken with great caution and under supervision, and also monitoring of the microbiome from time to time.
even, ma'am, nowadays they are doing the fecal transplant for this treatment or in correction of gut brain excess when this is deranged. So that is another modality people are using nowadays.
Yes, yes. In some other of my nutrition talks, once I remember your sports department had also arranged something where I had said that, too much of a good thing is also not good, and ye dil mange more is not right. And suddenly I've seen a question here regarding intermittent
Yes.
fasting. Yeah. So just like farmers leave their soil fallow for some time to allow for replenishment of the nutrients, similarly, we should also not overtax our gut. So somebody asked, intermittent fasting followed by, if I may say, feasting has been, Lent is going on, soon Easter will follow. Ramzan is around the corner and Navratras are just a few days ahead. So in many cultures, these kind of intermittent fasting protocols had been there to see that when seasonality changes, the microbiome changes, the diet changes, our body rhythms can change. The rhythm is not only a day night rhythm, we also have seasonal rhythms. In animals this is much more. We humans, with our behavioral interventions with ACs and central heating, have caused certain modifications, but the rhythms still act on us, and thereby there is a lot of wisdom in having a good mix of not disrespecting or thinking these are superstitious practices, but respecting all systems of medicine and joining hands and giving the world a scientific evidence. That's what I would like to say. Yeah.
My other question is, did you find any association between spices and cognition?
Yes, a very interesting question. When I had done my studies on spices, we had not found we had not tested for cognition per se. The cognitive lab I got as an adopted child. One of our faculty members had to leave for the Gulf and I was the co-guide, and thereby came my interest in that. But we had done studies on rat models with 2 summer trainee students, where we had seen that yes, locomotor behavior increases or improves, guided or target oriented, that we had done in that cage where we monitor the locomotor behavior, and also metabolic rate increases. But at the same time, some spices can also lead to what you call hallucinations, especially maize and nutmeg. But usually most spices, if taken in physiological to supraphysiological doses, taken in the way they are traditionally supposed to be taken, the method of preparation is also there. They actually help both for mental well-being, cognition, and physical health. They help in losing weight, or what we call optimizing weight, actually. So that is what I would like to say. But at the same time, during COVID, we learned that somebody was having CADA, TDS, or by the clock. So that kind of greed, of getting instant benefit, that doesn't work with such things. Traditional nutrients and including phytomedicines have to be taken in a very, I don't know the exact English word, but in a very sahaj way. That means the way they were intended to be taken in the amount they were intended to be taken. But yes, they are associated with cognitive advantages, and that is why I was using the word nootropics or cognitive enhancers. Yes. I can't hear you, Dr. Manjula.
I said, thank you, ma'am. It's a
Thank you
pleasure
so
to have
much. It
with
was a
us.
pleasure.
It was a
Yeah.
very interesting, very informative talk.
Yeah. You can please share my email with the participants. Thank you.
Thank you.
Thank you so much, Professor Kocher. We'll break for lunch now. I request all participants to rejoin by 1:30 p.m. for Professor Rajiv Anand's presentation, please. Thank you, and see you at 1:30 p.m. sharp.
My junior batchmate asked me to speak in MAMCO's Silver Jubilee. We celebrate every year on 20th of December. That what has changed in neurology. So first, it was a very difficult thing for me to conceptualize how to make this talk. So then I thought of how I started my neurology. In 1989, I started my junior residency in Department of Neurosurgery. So between 1989 and 2022, there has been a tremendous change. And that change has been driven by people behind the scene who in the lab conceive an idea. They try it on animals, and then they try it on humans, and then give it to us to practice and make people's life better. So my presentation is not like going to be pure science, which you people do. We are far away from them, but we practice that science to our patients' advantage. And for that, we are always thankful to you. Every talk for me is an opportunity to learn. And when I heard earlier speaker also, there is always something which you carry back home. So I was looking for Watson and Crick's quotes, and this is one of the quotes which Francis Crick wrote. He says, there is no scientific study more vital to man than the study of his own brain. I think which is the concept of our seminar which we are holding. Our entire view of the universe depends on it. So what is there in my mind, I see the universe like that only, and then I conceive things by my brain only. So I don't think there can be a better quote to describe than this to what we are doing. How the neuroscience has changed? We all know there has been tremendous advances in different fields. And those fields, in fact, even computer owes its duplication of how it is going to function by brain circuits. Because the first computer I used was during my MD thesis in Delhi University, which only had binary language. So you had 0 and 1 to be punched out. But then it learned parallel processing. So that is what happens in the brain. Brain simultaneously works at different dimensions. Today morning, in one of the speaker was asked, what is the role of dorsofrontal lobe in pain? So pain experience is actually, so pain is, we know a pain pathway, which is from periphery to spinothalamic tract to the parietal lobe to the thalamus. But brain does not work in isolation. So experience of pain is going to be by the frontal. All the emotions attached to the pain will be by the frontal lobe. So I think nothing in brain happens in isolation. So that's why now we are more into computation. So as explosion of new technologies has enabled discoveries that have fundamentally changed our understanding of brain function. So previously we were talking of focal brain areas which are controlling speech. In fact, I happened to go to a Yale computer lab where a scientist who was doing PhD in collaboration with Stanford. So he was doing research with Stanford doctors, and he told me what anatomy and physiology you have learned is not the anatomy and physiology when we study the function of the brain. And now we know that even though there are focal areas, but it is in association with other area that the whole function is depicted. So much of this work is based on animal experiments, which offer the cellular resolution required to understand system level mechanism. So there is switch from studying whole brain areas or a single neurons to studying intermingled neural circuits at cellular resolution. So we have a technology advancement which allows us to do it. There is increased focus on computation at the level of emergent properties in the large neural population. Many of these terms are probably better understood by you scientists, but I am also trying to understand. So practically, all this to me means that you have focal areas and you have those focal areas interconnected to whole of the brain. The transition from measuring neural activity in highly controlled or restricted setting to ones that accommodate ethologically relevant behaviors better. So I'll show you a slide. This is from Lancet, January 2022, which is actually dedicated to what has happened in the previous year. So this is a very good journal issue, which is totally dedicated to what have we achieved in last few years. So this is what is the opening, this is what the opening editorial is, that we were studying whole brain and now we are studying circuits. So we are concerned with neural networks. And now we know that the function is not achieved by whole brain, but it is the networks which are important. So we were studying cells, but it is not the cell, it is the connections. So turning properties of single cells, we were tuning to that. Now we are tuning to the properties of neural networks. Of course, you better know the techniques which you use in the labs. So this is what we were doing experiments. We all heard of Pavlov experiments. So these were designed tasks, but now we are studying behaviors in natural environments. Of course, this also means that we have techniques to monitor now. We have, like in neurology, we use telemonitoring, video monitoring for EEG. So we can do a EEG in patient's own environment. So it has all been held by your science, which has been brought from labs to our clinical applications. So this is how, so if I tell you about this, this is the pneumoencephalogram, very painful procedure. I used to do it in 1979 when I used to inject air into the spinal fluid and see the picture of air in the brain. So these are dilated ventricles in the brain. This was the only way. We didn't have CT, we didn't have MRI, but we had this knowledge. If I put the air from below, it will go into the brain. But it is a painful procedure. And this is the myelogram which we used to do. And see how the technology has changed. Now we have CT. So we don't need to do those things which were traumatic, painful. Then we have MRI and we have functional nuclear imaging. I'll tell you how it is evolved over years to make an impact in our clinical practice. Beyond this, now we are talking of advanced sequences of the same imaging, and we are also talking of brain initiative, which is going to be a further advance in understanding brain. So brain initiative is a brain research through advancing innovative neurotechnologies to produce dynamic picture of the brain that show how individual brain cells and complex neural circuits interact at the speed of thought. So this is actually when I'm taking a lecture for you. So we are now developing technology to monitor what is happening in the brain. At small level, this technology is helpful because if I have a functional imaging when a neurosurgeon is operating on brain, so if he can spare a language area, then it is useful for the patient. So patient is asked to speak and whatever areas of the brain lit up with that technology, he will spare them. So it's a big, big thing, which is pure science at the moment. But I feel that very fast pace, we are approaching it to make it clinical. So production and studying of connectomes. As I was telling you, we are now talking of connection. So if we were talking of speech, and when we were talking of speech in right-handed people, we will choose to think of temporal lobe. But speech is not merely speech. Speech is a connection between what you see, how you speak. So probably whole of the brain will be involved. So that is what connectomes are. So these are comprehensive maps of neural connections which can be seen by scientists when a particular function is being performed. And the tools used are diffusion MRI and 3D electron microscope. So science actually is moving at a pace for which probably all of us have to be very specific in our field, because you cannot be doing everything, but probably that field will come to me as a clinician, and in a particular disease, I'll be able to use it. So there is another facility we use, magnetometers, where we measure the brain activity by magnetic field. It's not MRI. It's a direct measure of neuronal activity. It's a functional imaging of the brain, not imaging, but functional mapping of the brain. This facility is available with National Brain Center, where they are using it to map the areas of patient which are important in finding out the focus of epilepsy. When the patient is not well controlled on drugs, then we think from where it is coming. So we can find out from where that is coming, and we can choose a better surgical area mapped by this. So why it is important? Because earlier I have to put in electrodes inside the brain. So that's an invasive procedure. This is a non-invasive functional way of measuring. It's just like EEG, but it is much more refined than that. So these are the technologies which are overtaking at a very fast pace. However, we all know that we have been overtaken by, even our technologies have not been able to so far conquer this pandemic, which is COVID. And so the Teacher of the Year award, this was on September last when I had to take a lecture. So we thought Teacher of the Year award goes to COVID-19. Why? Because it has taught us what life is about, simplicity and spirituality, and plus uncertainty. So every moment of your is the real moment. Rest are unpredictable. And as all of us know that some new strains are also coming up. With this, I will also talk about how the COVID has affected brain or neurological system. So I will not go into the details of this slide, but what I'll explain you is, so what does COVID do? So from wherever it enters the body, and the COVID incites a immunoinflammatory coagulopathy. So it is those phenomena which are responsible for symptoms, and it is a systemic disease. Like in body, we say it can affect any organ. In brain, we say it has a multi-axial involvement. Multi-axial involvement means it can affect the brain, it can affect the spinal cord, and it can affect the nerves also. And how it affects, it can cause direct involvement of the brain, or it can coagulate, it can cause coagulation in the blood vessels supplying the brain. So what we will get, we can get strokes. And as we all know, the loss of smell was as specific for COVID as RT-PCR test when the first wave of COVID came. So because the olfactory neurons were specifically sensitive, and patients used to report loss of smell and taste also. In fact, post-COVID also, people have not anosmia, but parosmia, where they continue to feel bad smell. So this is what the COVID is. Most of us are aware. Most of us would have had patients who are known to them who develop different types of complications. In neurology, we got almost every patient. We got strokes, we got Guillain-Barré, we got memory loss, ataxia, convulsion, delirium, loss of cognitive function. And this is what has been the story of COVID so far as far as the neurology is concerned. So first was anosmia and encephalopathy. Those patients who went to ICU, almost all of them had delirium. They may be because of hypoxia or direct inflammatory situation in the body causing encephalopathy, or there could have been certain areas of the brain which were affected by coagulopathies. So this was a multi-axial involvement, multi-pronged attack on human body by different mechanisms. And I personally feel we still don't know the precise answer to most of the things, because some of the patients continue to have strokes despite being on full dose of anticoagulation. And we also have to deal with the, so it's a immunoinflammatory coagulopathy, which is different. So you have to target the immunoinflammation. So many of the, like we have thrombotic, thrombocytopenic events by vaccine where the drugs are not anticoagulation, we give immunoglobulin. So understanding the mechanism has helped us to help some of these patients, but the final answer yet to come, and I think people are busy. And my hope for COVID is that we have a polyvalent vaccine once a year. Whatever is the strain, it should be effective, like we are taking flu shots. So this is in summary what we saw. I have already told you we had systemic things which caused coagulopathy, inflammation, and hypoxic damage to the brain. Then we had specific viral encephalitis, meningitis, myelitis. Isolation of virus from the CNS has been far and rare. Some autopsy studies have told us that there was a virus in the olfactory neurons. However, we did find markers and we did find signs of inflammation, and we did find coagulopathy in small vessel. We also had post-infection ADEM and GBS. Then there is peripheral nervous system. I had patient who had myopathies also. So this is another thing which is going to linger, and it is still lingering on. And almost everybody would have encountered patients of COVID which are still complaining of fatigue, headache, insomnia, vertigo. So encompassing all organs of the body. And initially people thought that it is just a post-COVID stress syndrome. It is not stress. We do find inflammatory markers high in certain of these patients. So this is another burden of COVID which we are dealing with, and hopefully with time they abate. So this is what I told you. Very rarely we have found olfactory neuron, but as I say, because this COVID was such a pandemic, the patients were isolated. So you couldn't have access to the patients and doing things. So only thing we have learned is that virus could invade the CNS via the olfactory nerve and possibly other cranial nerves. So this has been learned in certain postmortem studies. Why the science was hampered by COVID was because it was such a dreaded disease and you couldn't go near the patients. So severe COVID-19 is associated with inflammatory changes in the brain and choroid plexus. According to new research, SARS-CoV RNA was not detected in the tissue samples, suggesting that neuroinflammation occurs without the virus entering brain. So this I was already telling you. So we have now understood this much. In this slide, I just want to show that rare neurological complication can occur after COVID-19 vaccination, but recent studies shows that such complications are much more common after SARS-CoV-2 infection. So if we have to think about complication and hesitancy about vaccination, the complications of COVID are proven and dreaded. Complications of vaccines are far rare, and now we know that if it happens, we can manage them also. You should be alert to it. So another topic which has also been affected by our technological or so all the topic which the researcher takes are the topic where we are finding difficulty. So dementia is one such field we do not have cure, or we do not even have controls over dementia, or we cannot halt. And it is very important, some or other member, as we have aging population, then there will be dementia. So in India, also, it was thought to be a disease of Western country where lifespan was more. So in our country also, increasing lifespan means that the incidence and prevalence of dementia in India is growing and is expected to rise dramatically in the coming decades. Another point why we want to know and do about dementia is in India, most of the population has burden of risk factors which are preventable. So if we focus on them, then probably we can delay and defer dementia, or we can almost say that most of the people with these risk factor control will not have manifestations of dementia in their lifetime. I also saw diets can also help. So these two pointer I want to tell that we are also going to face a large burden of dementia because of increasing lifespan in our country, but we can do a lot about it by focusing on preventable causes. Of course, there is research going on. I'll also tell you how patient can be of dementia, but there are certain dementia where if properly technology is used, you can help the patient. So all dementia may not be degenerative dementia. There may be treatable dementia. So this is a patient which I recently saw about 3 months back. This is a 58-year female patient who was very active. She was running a travel agency, and she had a subacute onset ataxia with cognitive decline 10 months back. Initial workup was negative. She was worked up in a very good institute. Her MRI was negative. She was not a diabetic. Her metabolic workup was negative. She had some improvement. Therefore, the doctors thought it could be a TIA from which she is recovering. Recovered, and MRI doesn't show any infarct. So she was put on aspirin and statins. But she continued to worsen and she had to stop her work. Her daughter, who used to talk to her mother every day or every week from UK, she found her mother incoherent on phone. And she rushed to India and brought her back to me, referred by one of my colleagues' son, who was her batchmate. So we found that since our MRI was normal, so this is how the technology helps us. So this is a FDG PET study of the brain. So since this patient had structurally normal brain, but functionally she was deranged. So I wanted to know that whether there is a hypofunctioning in the brain or a hyperfunctioning. So I ordered for a FDG PET. So if you see, she had ataxia. So she has a high uptake in cerebellar region. So she had a very prominent feature, dysarthria, ataxia, all features of cerebellar sign, as if you are a drunken man. Then she has a parieto-temporal involvement also, and these are basal ganglia. So what we found was that we had a functional localization of her symptoms, which we were clinically seeing. The another thing we found was we are not finding hypofunctioning, we are finding hyperfunctioning. That means it is a encephalitic cause, whether it is an autoimmune encephalitis or an infective encephalitis. So this gave us lead how to treat this patient. Of course, we worked her up and we ruled out any infective cause, and what we found was very high titers of anti-GPO positive antibodies. The level was around 650. So this is a autoimmune Hashimoto's encephalopathy. So this is a treatable dementia. What we did was we gave her pulse methylprednisolone for 5 days and then gave her tapering steroids. 2 months follow-up, she's back to her work. So this is what properly used technology given to us by the scientists can help the patient. So this is how we utilize all the investigation which are available to, but you have to be aware what you are looking for. So there is a quote which says you always find what you look for. So I went for a functional imaging because the structural imaging was normal, and functional imaging also told me it's a hyper functioning. If it was hyper functioning, I would have thought of a degenerative disorder and left this patient alone. So this is all research where we are trying to find out that what markers are cause of degeneration, so that someday we can find. So I will just highlight one. The inhibition of noradrenaline transport of apolipoprotein can induce the aggregation of tau protein, which is supposed to, if cumulated, is the hallmark of Alzheimer's. The finding provide insight into effect of APO gene on Alzheimer's disease risk. So we have people in labs who are working on this thing, and if they find an asparaginase endopeptidase which can reverse this effect, probably we can find a cure. But these are efforts which may someday bring light to the patient's life. Another thing we know is every patient of Down syndrome, almost 90% of them will have Alzheimer's like pathology. So there was not much focus on that, and now we are trying to find out novel biomarkers of aging and dementia. bring new hope to this medically vulnerable population and can also help them. So this is how the bridge between say research and therapeutics help us. This is another thing. Almost all scientists are now talking about deep learning distinguishes tauopathies. So this is deep learning model is being applied to almost all types of science, and this is also being applied to dementia. So this is another disease, which is a degenerative disorder. So as I told you, the scientists help us by focusing on those diseases which have no cure. Of course, Parkinsonism, we can modify the symptoms, but we don't know how to reverse Parkinsonism and how to effect cure. So there are now people are finding, so there is a brain lymphatic and glymphatic system alteration have been found out. So the findings suggest that dysfunction of brain drainage pathway represent both an early biomarker and a potential therapeutic target for PD. So these are the research initiative which I happen to read because of you people, and I find them very exciting and interesting because tomorrow we may be able to use them. So we have another group of diseases which are mitochondrial myopathies or mitochondrial myopathies affecting all organs of the body. So myopathy, encephalopathy. So those can also be diagnosed by, you understand more than me, those who work with genetics, that applying genomics, we are specifically able to diagnose and we can have advances in mitochondrial medicine. So far, we used to just diagnose them clinically and then do some biopsy study to find out mitochondrial myopathy. We will rarely do the biopsy of brain, but we can now do genomic studies to diagnose them. Of course, we don't have much treatments available even as yet. So mitochondrial medicine is poised to benefit substantially from increasing use of RNA sequencing of tissue samples. Advances in pre-processing and sequencing of transfer RNA are enabling new insights into the molecule, which plays an outsized role in the etiology of mitochondrial disease. Thanks to COVID, we are all talking about RNA nowadays. So most of us are getting familiar with this part of science also. This is another thing why the migraine is more common between female. We all know that it is a hormonal link of female hormones are more prone to cause more frequency of migraine. However, prolactin signaling amplifies the ability of calcitonin gene related peptide, which is a peptide responsible, and we have now drugs to monoclonal antibodies against this peptide to induce migraine in females, according to a new study in Annals of Neurology. The finding in mice could explain the sex difference in migraine prevalence in humans and might provide new targets for therapeutic intervention. I always say that science will lead to better therapeutic intervention, and that is what we are looking for. Again, so it is not only that migraine is pain, so it is pain modulated by hypothalamus, and in this hypothalamus loses control in migraine. That is why it cannot prevent that process, or it cannot stop that circuit which is self-propagating to cause migraine. So they say the research supports the idea that the hypothalamus is central to migraine pathophysiology. As a student, we have learned vascular and neurogenic approach. Whether it is a cortical phenomena, whether it is a vascular phenomena, but it is much more beyond that. And at molecular level, we can earn more precise cause of migraine. This is another disease, amyotrophic lateral sclerosis. Those who are familiar with it, Stephen Hawking was the one who was suffering from it. So there is no cure as on date. So we are now learning that non-neuronal cells in amyotrophic lateral sclerosis may have a role, and why we are learning them is important because treating those non-neuronal cell may have some basis by which we can modify the disease. So human-induced pluripotent stem cell permit the investigation of non-neuronal cells carrying ALS-associated genetic mutation, but multicellular co-cultures might be needed to entangle their nuanced interaction with motor neurons. So this is how we may understand, and this is how we may be able to change it. Human biofluid markers derived from non-neuronal cells offer an important window into the in vivo pathological milieu and show potential as early markers of therapeutic response. So based on this, we have an experimental new therapy for ALS, AMX0035 and sodium phenyl butyrate, which targets endoplasmic reticulum stress and tau ursodiol, which targets mitochondrial dysfunction. So these two drugs individually exhibit neuroprotective properties in experimental models of ALS and are expected to have additive or even synergistic effect when used in combination. So these are the things which excite me, and probably some of you may find them to be working in your lab. So this is another topic which thought I will share with you. So when people get epilepsy at old age, they are more prone to cognitive decline. So what you people have helped us to know is why it happens. Why it happens is late onset epilepsy of unknown cause have been shown to have amyloid pathology in the brain with amyloid beta deposition, increasing their risk of developing cognitive decline over decades following seizure onset. So there may be a link between late onset epilepsy and early dementia developing. Seizure can be harbinger of dementia, and identification of people at high risk of cognitive decline at epilepsy onset might allow crucial interventions early in A beta deposition, thereby preventing further neurodegeneration. So this is again what is I was talking to you about seizures and cognitive decline. Now it is headache, and people with epilepsy also has a basis. The lifetime prevalence of migraine is 52% greater in individuals with epilepsy. Before reading this slide, I'll tell you that this is because migraine and epilepsy now are many of the syndrome based on channelopathy. They may have one genetic marker and they may have many phenotypic marker. So with one channelopathy, you may have migraine and epilepsy together or separate. The symptoms of epilepsy and headache can present diagnostic challenges. Of course, you have to do EEG, and there may be ictal headache, means even epilepsy may present as headache. So enhanced neuronal excitability might be the mechanism linked between headaches and seizure. Several genetic mutations can cause epilepsy and migraine, but the genetic association between polygenic form of epilepsy and migraine remains unclear. Novel therapies include CGRP blocking drugs for migraine and neuromodulative non-pharmacological approaches for migraine and epilepsy, that is behavioral and self-management approaches, are increasing in popularity. This is because we are finding that hypothalamus, which can be affected by behavioral modulation, is the seat of origin of migraine. It may also have effect on epilepsy. This is another disease for which there is no treatment available as. So I am presenting to you where the work is going on in the labs to find out cure for incurable diseases. Spinal muscular dystrophy, AR mutation in gene SM1 and gene SM2. SM is survival motor neuron protein, which is insufficient to maintain the health and normal function of motor neurons. The number of copies of SM2 varies between individuals, and some patient might have the same number of SM2 copies but different. This disease is also like amyotrophic lateral sclerosis, I told you, but this is from the childhood. Then adolescent and adult variant of this disease are there, and this is slower progression. A motor neuron disease progresses between say 2 years to 4 years, maybe the maximum lifespan, but here it is a slow progression of muscular atrophy, and we are trying to find out what are the cures for this. So one of the drug which is almost exorbitantly costly, which is available for spinal muscular atrophy type 1, which happens when the child is born, and if you start the therapy, probably you can reverse the effect of spinal muscular atrophy and child can live normal life, but the cost is in crores. So this drug was available in AIMS as a part of this study to make it available or test in Indian population. So one of the first drug was nusinersen, the first approved drug for patients with spinal muscular atrophy, promotes the inclusion of exon 7 in SMN2 transcript so that it can be translated into functional full-length SMN. So we have dysfunctional, so it changes that into functional, so which allows motor neurons to survive. The drug is administered by repeated intrathecal injection, and this directly targets motor neurons with efficient delivery to cortical regions of the brain. So this is available, but practically very costly and very difficult to administer. Whereas the other drug, which is still not been approved, is risdiplam. It is an oral drug. It provides opportunities and challenges. The safety profiles and their easier routes of administration, so it can be taken orally once daily, acting on SMN2 and given a single or there is another drug which can be given as a single intravenous infusion in a lifetime, which acts on SMN1. So these are appealing new scientific discoveries which can change the life. Now, the another very exciting field which has changed over the years during my practice is acute stroke treatment. So we didn't have anything to treat in a way of stroke, but today we have a better diagnostic facilities in the form of CT, MRI. We are able to target the clot in the blood vessel, which is going to cause ischemia. So if I have a patient between 3 to 4.5 hours, I can anticoagulate. Then we can anticoagulate non-valvular AF. We have evolution of stroke ICUs. We have team approach. We have preventive strategy and rehabilitation, which has made the life of people who suffer from stroke better. So this is how, this was a patient who was operated for a carotid body tumor in the hospital in Delhi. And he post-operatively, when he woke up, in carotid is in our neck, and carotid body, that carotid artery goes to the brain and supplies the brain portion. So in this patient, you can see that this right side of the brain, this white is a DW weighted image, which already shows that he has developed an infarct. The problem was we did not know at what time it developed, and it was a post-surgical patient, so we could not give an injection of thrombolysis. So what could we do? So if we leave this patient, so this patient was diagnosed with it when he recovered from anesthesia. Surgery lasted for about 2-3 hours, during which they do clamp the carotid for some time. So this was a result of that. So what happened? So what happened was, so this patient was taken up for a procedure which is known as thrombectomy. So this is a catheter which goes inside the vessel, and this is a device which takes out the clot. And so this is actually what happened. This is a carotid bifurcation where there was a total block. So microvascular catheter was passed, and now you can see that post thrombectomy. So they took out the clot, and you have another run of dye which is going to all vessels of the brain, which is open. Think of a possibility: if I would not have done this, then this patient would have lifelong. He's a 24-year male. He would have lifelong disability of left-sided weakness. So thanks to the people who are giving us imaging technology, who are giving us interventional technology, so that patient can be brought back to normally. This was the clot which was there inside the brain vessel, and when removed, the patient post-operatively only had this small infarct, and he was able to go back home. What is happening in last 1 year is, in the world, they are not now talking, I don't know when it will be in our country, but here you see the image of a helicopter. So they're not talking of waiting in the hospital for a patient of stroke, because we say time is brain. So each minute we lose about 20 lakh neurons. So whenever they want to target the patient wherever it is as soon as possible with mobile CT scan van or mobile stroke van, which can prevent that wastage. Of course, it is utopia for a country like ours, but this is what they are trying to do as an advancement for this thing. Another thing I'll talk to you about is multiple sclerosis. One thing which has happened in MS is we have better diagnosis now. to think of multiple sclerosis and diagnose it as a first disease in my undergraduate days was getting failed by the examiner. We never used to think that multiple sclerosis exists in India. For similar symptom, it will give a cause like tuberculosis or other thing. But we know that we can now, with the technology being available, we see a lot of patients of MS, and there are two things. One is a MRI, clinical examination of the brain, MRI of the brain, electrophysiology, and CSF. Don't go into the details because we will not be able to finish. So, but what has happened is we used to think of wait and watch. Now we think of early treatment delays disease progression and leads to better outcome. If I wait and watch, my patient will have an attack and he will have a permanent deficit. We are also not targeting right therapy for the right patient, because we have now many drugs available, so which were not available. Not even a single drug was available when I was trained into neurology between 87 to 89. We were using vitozentron, we were using methylprednisolone, but specific drugs were not available. But today we are talking of personalized medicine, because we can precisely diagnose the patient, what is his level of disease and what type of drugs will be effective. So this is how the drugs have evolved, and we are still getting more and more drugs being made available to us specific to this disease. And then you see here we are talking going up to even 2025. Another big thing in this disease is to cure this patient by stem cell transplantation. So I'll not talk to you about. So early diagnosis, treatment option, prognostic factors, and better outcome. This is how it is. We have B cell in multiple sclerosis. We all know it is the predominant cell which causes immunoinflammation and damage to the myelin. That's why the people get. And recently you have articles that Epstein-Barr virus may be responsible for this initiation of activation, and now we have a lot of B cell therapy, and we have cell depletion treatments also, which are both by the drugs and also by the stem cell transplantation. This is a very exciting thing. So there was one fortuitous intersection between multiple sclerosis and COVID-19 in 2021. The report of an mRNA vaccine targeting myelin oligodendrocyte glycoprotein. This vaccine triggers regulatory T cell development and suppresses experimental autoimmune encephalitis induced in mice, either by a MOG or other myelin peptides, suggesting bystander tolerance. So let us see how exciting it could be. So wouldn't it be remarkable if the therapeutic progress spurred by the COVID-19 pandemic was also marshaled to treat and possibly even cure multiple sclerosis? So if we use the same mechanism, so as I told you in the beginning, so the Teacher of the Year award goes to COVID-19. So our scientists are very smart. They might use this opportunity to give us a vaccine which can cure multiple sclerosis. This is about epilepsy. So see, I started practice somewhere here and we had only 2-3 drugs available, and now we have more than 20 drugs available. And we have other things. People who do not respond, we can do diet and surgeries and they can help the patient. This is a new thing which I have learned. I have not seen being practiced in India. So this is a without surgery, you will just target a laser. Of course, you will identify the lesion which is causing surgery, which is causing seizures, and then you can treat the patient. So Visualers is a probe inserted in operating room, transferred to MRI, laser treatment. MRI monitors the safety of laser. It's a real-time probe removed. Patient can leave the same day. So there are neuromodulatory treatment. So we will now, I think. So what epilepsy care look like in future? Personalized medical choices based on genetics, through anti-epileptic treatment and treatment directed right at the seizure focus. So this is how we are being helped. So neurogenetics is another exciting field in last 2-3 years, because whole exome analysis is available. Many diseases which we were not able to diagnose, we are able to diagnose. And once we are able to selectively diagnose, scientists are attempting to treat them also. So I'll skip and I'll come to this slide. So examining disease, we gain wisdom about anatomy and physiology and biology. But this cannot happen if I don't have the knowledge of molecular medicine, or if I don't have the knowledge of what I am looking for. So in examining the person with disease, we gain wisdom about life. So all this happens. There is another quote. So I always find what I look for. So I need to have a, you can say, AI-driven knowledge into my brain, what I'm looking for in my patient. So this is an important quote for me. But this is even more important for clinician. People don't care how much you know until they know how much you care. So this is still relevant to our field and your field also, because you represent those people behind scene who are trying to make difference in people's life by telling us what will make the difference. Thank you.
Thank you, Dr. Rajiv, for your excellent presentation. I have some questions. I have myself worked with mitochondria, so I was intrigued about the role of mitochondria in these diseases. You explained about the expression of tRNA especially. So the mitochondria is involved at the level of expression only, or mutations or SNPs in mitochondria, or their copy number variation. Like all these things are important, or the known protein coding tRNA, rRNA, they are more involved?
First, I will admit that I am not the right person to answer this question, but clinically, I will tell you, we have certain mitochondrial disorders. So if you are asking me, probably everything is true. What you are asking me is at all level, it may be true. There are varied types of mitochondrial disorders and there are varied manifestations. So what we have been able to now do is, we have been able to diagnose them by the probes or by the mechanism which you have brought up in the lab, but we have not been able to change them. So probably I am not the right person. Some scientists can answer this question. And if there is somebody, I think I would like him to join in and tell me about this. Because as I told you, I am very excited by what you are doing in labs. And all this is not my work, it is your work. But I am also very excited how I should use it in my practice. I have diagnosed, we give so I had a patient say of MELAS, mitochondrial encephalopathy, lactic acidosis, recurrent strokes. So probably if you see, the mechanism will be, what you are talking is what is a manifestation, and behind that mechanism will be multiple. Like today morning, some question was asked about what is central sensitization, what is hyperalgesia, and what is allodynia. Right. So actually central sensitization is the mechanism for hyperalgesia and allodynia. They are not same. Similarly, what you are asking, when person gets more knowledge, he will be able to segregate and tell you precise meaning of each thing, but they will be important. So like central sensitization leads to hyperalgesia. Hyperalgesia is when you feel a painful stimulus, more pain. And allodynia is when non-painful stimulus causes you pain. But both are caused by central sensitization. Similarly, mitochondrial disorder, the way I am talking is central sensitization. I am talking in a holistic manner, but if you want to split them, you will have to talk to a mitochondrial expert.
Okay. I have another question. Like, what is the mechanism for brain fog in COVID patients? So I read a lot about brain fog during the COVID times.
So this is a very important question. So, and this is a question partially has been answered. So as I was alluding to you, what are the symptoms which linger on after the brain fog? So, and why they are lingering? So most of the time you are seeing it in those patients who had severe COVID, but it is also known that those patients who did not have a severe, who had mild or asymptomatic COVID are also having it. What it means is brain fog is a symptom which patient reports. So he says, I feel as if I am not able to process the information or I am not able to concentrate. So that is a layman term. Why it is happening in the brain? Because as I told you, there may be persistent inflammation in the brain still left, as we have understood that COVID-19 is a immunoinflammatory disorder triggered.
Right.
So, and it is not immunoinflammatory. So it may be immunoinflammation. And when you see clots, then I call it immunoinflammatory coagulopathy. So all these factors are important. So people are having persistent, like you have a something like we were talking in the morning. We didn't talk about chronic fatigue syndrome. We didn't talk about fibromyalgia, which also are responsible. So there are many things which happen and which cumulatively add to. Suppose if a patient has very low energy, and those patients whose lungs have been damaged may have brain fog because they may not be able to give them enough oxygenation. So it's a multifactorial thing. But in totally recovered patient, what I do is I do the inflammatory marker again. So if some people have persistent high level of inflammatory markers, in them probably you can have an evidence that inflammation is still going on.
Okay. So, I mean, there is a connected question. So can these viral pathologies, like in case of COVID, serve as a model for understanding these neuroinflammatory roles in neurodegenerative disorders? Like,
Yes. So the one of the point I raised was, so they found that Epstein-Barr virus may be a trigger for multiple sclerosis. This is 2020. This is the 21. So this has come, almost anybody who subscribe to any science, it comes as a news or as a paper. So so I have a series of 450 patients of chikungunya, out of which I had 52 patients having neurological complication. If I see chikungunya, if I see dengue, if I see COVID, there are two things which we face. We face a acute infection, and then we face a post-infections. So when there is a post-infections complication, they are triggered by some breakthrough in the patient's autoimmunity status, because then we are getting myelitis, we are getting delayed complication, which are triggered by the same virus breaking our autoimmune system, which is protective and it becomes destructive.
yeah, amazing discussion. Nice talk, sir. Any other question? There is nothing in the chat. I think we are done with it. We can move for the next presentation. So Imran, you can unmute yourself and start.
Thank you so much, Professor Anand, for this brilliant talk. The next presenter in this post-lunch session is Professor Unup Gogia. Professor Gogia is the head of the Department of Anesthesia at ESIC Medical College and Hospitals in Faridabad, Haryana. He'll be speaking on the advances in measurement of depth of anesthesia. Professor Gogia's research subjects are anesthesia and emergency medical care. Therefore, his presentation will elaborate upon the latest developments in the measurement of the depth of anesthesia, as gauging the depth of anesthesia is important to prevent intraoperative and post-operative sequels. Welcome, Professor Gogia.
Professor Gogia, you are co-host now, and I have your slides. I'm going to present them, and you can just start. Professor Gogia, you are co-host now. You can unmute yourself. I think there is a technical glitch. Please wait for a moment.
Can you listen to me? Hello?
Yes, you are audible. Yes.
But I cannot see the slides. Okay. This is the presentation. Can you see the presentation?
Yeah, I've shared. Yes.
Oh, can you see the presentation now? Hello?
Yes, sir, it's there on the screen.
Okay, okay. Thank you very much for giving me this opportunity to speak to this august audience. My talk about of today is about the neurological monitoring and depth of anesthesia. It is in two parts. First, I will take the depth of anesthesia and then followed by the neurological monitoring, which we do during the procedures. So what is depth of anesthesia? Actually, anesthesia is a drug-induced reversible condition. It is composed of four physiological behavioral components like amnesia, analgesia, immobility, and stability of physiological systems such as cardiovascular, respiratory, neurologic, and autonomic systems. Nowadays, they have combined amnesia and analgesia together and they call it narcosis. So these three things, narcosis, immobility, and reflex suppression, these three things parts the basic, they form the basic part of anesthesia, and it is called modified Gray's triad or triad of anesthesia. So this we achieve with the use of some induction agents like thiopentone, propofol. These are the drugs by which we put the patients to sleep. We maintain it by using the muscle relaxant, which produces the immobility, and some amount of inhalational agents. And the reflex separation is achieved either by use of muscle relaxant or by use of drugs like narcotics, inhalational agents, and sometimes the local anesthetic block. So the whole three things, they form the basis of anesthesia. Now, what is awareness? Awareness is the explicit recall of events when patient is under general anesthesia. Depth of anesthesia is important to prevent the awareness and prevent perception of noxious stimuli intraoperatively. This is difficult to assess objectively as amnesia is measured indirectly by the extent of consciousness. This is also dependent on the intensity of surgical stimulus and the drug dosage which we use. So why it is important to monitor the depth of anesthesia? The aim of monitoring depth of anesthesia is to maintain amnesia and anti-nociception intraoperatively by titrating the drug dosages while we maintain the stable physiological function. Actually, anesthesiologist is the physiologist to the surgeon. So what are the advantages of measuring the depth of anesthesia besides the intra-op homeostasis of the patient? You can use optimal intraoperative drug dosing, which will prevent awareness. Also, optimize the hemodynamics during the surgery, and titrating of drug dose also leads to an early recovery following general anesthesia and prevention of problems such as post-operative cognitive dysfunction, especially so in elderly. According to a multicentric US-based study conducted in 2004, the incidence of awareness during anesthesia was 0.13%, or 1 or 2 per 1,000 patients, with 0.024% patients having possible awareness. So it was minimal, but it was still there. Now, the factors which increased awareness were found to be higher ASA physical status 3 and 4. Now, just for your knowledge, I must tell you that we classify the patient according to their medical condition into different status. Grade 1 is the one where patient is fully fit without having any evidence of disease. And grade 4 is the one with who has a severe systemic disease, which is incapacitating. So in between, the category 2 and 3 occurs. So when the patient is of higher ASA grade, then there are chances that such patients are kept in lighter plane of anesthesia with minimal of drugs. So the procedures which are performed, which lead to higher incidence of the awareness, are cardiac, abdominal, thoracic, ophthalmological procedures, and of course, cesarean sections, where we deliberately keep the patient very light till the delivery of the baby. And also contributing to this are sometimes the equipment misuse or technical mishaps, which can lead to a awareness during anesthesia. Now, what are the problems which are associated with awareness? There is a significant psychological impact such as post-traumatic stress disorder, nightmares, sleep disturbances, and anxiety. This is common in post-operative period in patients who have the problem of awareness. So, how do we measure the depth of anesthesia? There are some physiological signs which we see every time. That is the loss of consciousness after induction. When we give the drugs, we see whether the eyelash reflex is gone or whether the contact with the patient is gone. We ask the patient repeatedly whether you can answer something, what is your name or this thing. And when the patient stops responding, that is first sign. Then rise in heart rate and blood pressure. This is due to nociceptive medullary autonomic pathway. If the heart rate and blood pressure are rising, we know that patient is in the lighter plane of anesthesia. Sweating because of autonomic nervous system stimulation, same way, lacrimation, pupillary dilatation, and sometimes movement, which may be masked by use of muscle relaxants. So muscle relaxants, in fact, they produce a state which is by which the patient appears to be anesthetized, but still may be in lighter plane. So that is why so many things have been devised, and these monitors are called now bispectral index, which is called BIS, patient safety index, narcotrend, entropy, and SNAP index. These are the different monitors which are basically EEG based, and they help us in measuring the depth of anesthesia. Besides the depth of anesthesia, these monitors may be of some use for identification of inadequate cerebral blood flow to the cortex, to guide the anesthetic-induced decrease in cerebral metabolic rate in anticipation of decrease in cerebral blood flow or raised ICP during neurosurgery, and prediction of neurological outcome after an incident. This is a very, very important field that in ICU, after a neurological injury, when the patient is recovering, if you are monitoring these EEG-based indices, then you can easily predict that the patient will recover, or the prognosis of the patient is better, or it is going to be worse. So what is this? EEG is a summation of excitatory and inhibitory postsynaptic potentials, which are spontaneously generated by pyramidal cells in cerebral cortex, which you know better than me, and they help in assessment of functional integrity of the neural circuits as well as the depth of anesthesia. So I will discuss one by one these things. Bispectral index. This was empirically derived EEG based. This was made by US company Aspect Medical System. It has an algorithm for index processes EEG in near real time to calculate a value between 0 to 100, where 100 implies a conscious awake patient, while 0 denotes isoelectrical EEG. So the appropriate anesthetic depth which is required for this index during anesthesia is between 40 to 60. So this is what we try to keep during anesthesia. We know that if the indices is between 40 to 60, then patient is adequately anesthetized. Neither he is too light nor he is too deep. So 3 types of EEG analysis are used: spectrogram, bispectrum, and time domain assessment of burst suppression. The lag period is about 20 seconds. For the most anesthetic agents, the EEG, how it responds to the anesthetic agent is that it becomes lower in frequency and higher in amplitude as the depth increases. So the frequency, it becomes lower, and the amplitude, it becomes higher. This is the crux of EEG monitoring during anesthesia. But there are some agents which are exception to these. These are ketamine, which increases the frequency. Ketamine, as you know, it's a agent which leads to corticomedullary dissociation, and patient may be opening his eyes, but he is still anesthetized. So it leads to a detachment from the surroundings, but it produces exceedingly good analgesia. So it is used sometimes for multiple procedures, small procedures like radiological investigations, and at the time of multiple trauma to patients, and sometimes for pain relief. Nitrous oxide, it increases the amplitude of high frequency waves and decreases the low frequency waves. Dexmedetomidine, this is agent which is alpha-2 agonist, which is very commonly used nowadays to promote the analgesia. It also leads to increase in amplitude of low frequency waves, leading to erroneously high BIS values, despite the patient being only sedated and not anesthetized. So these are the 3 exceptions. Otherwise, most of the anesthetic agents will have lower frequency and higher amplitude of the waves. That is how the electrodes are placed. It is put on the forehead and one on the temples, and you can see on the monitor it gives a singular value of 64. Here it is shown in the slide, which shows that patient is almost well anesthetized. It's not light enough. If it is below 40, patient would be light. If it is more than 65, then we know that the patient is slightly on the deeper side. What are the factors which affect BIS? Anesthetic agents, we have already mentioned some of them which are exceptions, like ketamine, nitrous oxide, and dexmedetomidine. Etomidate, it is an induction agent which produces myoclonus, which induces a temporary rise in BIS. Halothane also has higher BIS compared to isoflurane and sevoflurane. Isoflurane and sevoflurane are the two more common agents which we use now for inhalational anesthetics. The baseline abnormal EEG, you can see, you can have a decreased BIS in post-ictal state, dementia, cerebral palsy, and severe brain injury and brain death. And increased BIS you see during epilepsy. So that is what I was talking, that even it has a use in post-operative recovery of the patient. If you are monitoring BIS continuously in ICU, then you can say after neurological injury that patient will recover or not, seeing just the trend of the BIS values. So this is a diagrammatic representation of the bispectral index, where 100 shows the awake and 0 shows the isoelectric EEG, 40 shows deep hypnotic stage, and 60 shows general anesthesia. Then there is another thing which we use very commonly and which is not EEG based, which is called MAC, which is known as minimum alveolar concentration of inhalational agents. This is the concentration of inhalational agent which will produce immobility in 50% of the subject population to a noxious stimulus like skin cutting. So that is called MAC. The MAC value can be read directly on our monitors nowadays. So this was used before BIS. So now there have been several comparisons with BIS versus MAC. One was Be Aware trial, where patient monitored with BIS had lower incidence of intra-op awareness compared to the other standard of care indices. Then there was Be Unaware trial, which was conducted after Be Aware trial, and because of the concerns with the designs in the previous trial, and it was found that there is no difference between BIS and MAC group. Then BAG recall trial, which was followed by Be Aware trial, BIS was found superior to MAC monitoring. So the general consensus is that if you are using BIS monitoring, then probably it is a better monitoring system than MAC alone. To tell you frankly, the BIS responds very beautifully to inhalational agents. Once you have induced the patient with the induction agents like propofol and thiopentone, once we introduce inhalational agents, the BIS goes down very steeply. But this does not respond so well to intravenous anesthetic agents like propofol infusion or infusion of narcotics. There it does not respond so well. So it is basically the inhalational agents with which we do the maintenance where response is seen. Then coming to the entropy, which is another system. Entropy basically means a disorder, means when the patient is awake, there are lots of stimuli being given in the cerebral cortex. So entropy measures the degree of chaos in the system. So it is a monitor which was developed by Datex Omida. It is there on our workstations, which measures the apparent decrease in entropy of the EEG signals. That once the inhalational agents are started, then the patterns become more regular as patient is put under anesthesia. So it measures two types of frequency. One is the response entropy, which measures changes in high frequency range up to 47 Hertz, and state entropy, which is which measures the changes in low frequency rate up to 37 Hertz. So as patient is induced, the response entropy decreases faster than the state entropy. So entropy also shows erroneous reading with ketamine and dexmedetomidine, because basically it is again EEG based. So that thing will be there. Here also, you can see the electrodes are applied on the frontal area and one on the temples, and it shows the value of response atrophy and the state entropy. So this is another method of measuring the depth of anesthesia. It also gives you a figure by which you know that sufficient depth of anesthesia is there. Then patient safety index. It is again proprietary algorithm process EEG to assess level of consciousness. It is very similar to BIS. The range is between 0 to 100, but ideal for general anesthesia is less than what values are there for BIS. It is between 25 to 50. Previous models had both frontal and occipital EEG montages, but nowadays we only do it from the frontal areas. It is not as widely used as BIS. So BIS is the one which is standard. Rest all are the corollaries which have been developed by different. Narcotrend, this is another monitor which has derived from a system developed for visual classification of EEG pattern associated with various stages of sleep. This was developed by the Germans. The aspect medical which developed this was from US, and it has again the electrodes are applied on frontal area, and it classifies the EEG in awake, sedated, light anesthesia, general anesthesia with deep hypnosis, and general anesthesia with increasing burst suppression. Alphabetically scaled translated into dimensionless index, scaled from 0, which is deeply anesthetized, to 100, which is awake. Reported mean narcotrend values after induction 72 to 80, and at emergence around 80. So there were comparison made between the BIS and narcotrend, and it was found that that BIS is slightly superior to narcotrend, because narcotrend is unable to differentiate reliably between conscious and unconscious patient during general anesthesia when neuromuscular blocking agents were used, and which is a common course during anesthesia nowadays. Similarly, SNAP index, this is another index which is EEG based. Low is between 0 to 40, high is between 80 to 420. The SNAP monitor was that it measures the higher frequencies also, but unlike BIS, which only measures the low frequencies. So use of these frequencies would theoretically improve the responsiveness during increased brain activity. So these are the 5 indices which we are using to measure the depth of anesthesia. And I show you the effect of anesthetic drugs on EEG, these are the different drugs which are showing the effect. Just remember that the common inhalational agents which we use, they decrease the frequency, but they increase the amplitude. Rest all are some variation, except some are the exceptions like etomidate, ketamine, and dexmedetomidine. So this table just shows that, but just remember what I told you. What are the other factors which affect EEG? One is hypoxia. Slowing of EEG is similar to ischemic insult. It is a non-specific global effect. So if patient has severe head injury and some hypoxia, then it will show a slight slowing of EEG. Hypotension, again, with this, the response would be similar to ischemia as well. Especially significance is under anesthesia, as you want that normal cerebral autoregulation should continue and it should not be disrupted. Hypothermia also leads to slowing of EEG with complete stopping at 15 to 18 degrees Celsius. We sometimes take the patient deep in hypothermia in some of the cases where we require the preservation of the brain during neurosurgery. It is not a very common procedure, but yes, for brain preservation, sometimes this is done so that minimal insult occurs to the brain. There also the EEG will show slowing. Hypocarbia, you all know that this is one of the factors which may activate the seizure foci. So these are the other factors which may affect the EEG. So what are the other methods to measure the depth of anesthesia? Isolated forearm technique, which was used previously, that was that a tourniquet was tied in the hand, and after the muscle relaxant is given, and after anesthetizing the patient was kept relatively little light and asked to press a bulb by hand, because it is an isolated arm technique. So that area is not is without the effect of muscle relaxant. So till what time the patient is able to do that? So this was one method to measure the depth of anesthesia. Then lower esophageal contractility, frontal muscle EMG, which is now part and parcel of most of the indices, heart rate variability, which I have talked about before, and minimum alveolar concentration of the inhaled anesthetic agent, which I talked earlier. So these are the other methods to measure depth of anesthesia. The only thing which I want to tell you that previously we used to do measure the depth of anesthesia by the physiological changes in hemodynamic, that the blood pressure is rising and the pulse rate is rising, then patient may be little light. But since there are other factors which can also lead to this, so depth of anesthesia was misinterpreted. But now we have a definite monitor by which we know that a single indices will tell you that patient is adequately anesthetized. And now if the heart rate variability occurs, or if the blood pressure goes up, then you know that some other cause has to be found. Then coming to neurological monitoring, which we do during surgery. These are called evoked potentials. These are the electrical activity generated in response to motor or sensory stimulus. This can be classified on the basis of type of stimulus. Sensory evoked potential, which can be classified again into somatosensory evoked potential, brainstem auditory evoked potential, or visual evoked potential, and motor evoked potential is another category. So these are type of evoked potential which are used for neurological monitoring. What are somatosensory evoked potential? This is the response which is recorded after stimulation of a peripheral mixed nerve. Most commonly, these nerves used are median nerve, common peroneal nerve, and posterior tibial nerve. And you have another set of electrodes which are placed at Hub's point or at the cervical vertebra level for the dorsal column nuclei, or on the scalp for sensory parietal cortex. The cortical short latency waveform most commonly recorded due to least effect of the anesthetic depth, and long latency waveform. These are the two waveforms which are recorded. So this diagram shows you how the positioning of the somatosensory. These electrodes are placed either in the ankle or in the on the common peroneal nerve, and sometimes the recording electrodes are put on the dorsal columns in the cervical vertebra, then in the occiput or in the parietal cortex. And this shows the arc, and these are the somatosensory evoked potential, which measures the impulses which are generated in the peripheral nerve and carried to the sensory cortex. So what are the types of SSEP? It is near field potential, where electrodes is kept closer to the actual site of signal signal generation, that is within 3 to 4 centimeters. And more commonly, far field potential, where electrodes are kept at a distance from the actual site of signal generation. Advantages are, as the electrode site is far from the signal generation, there is a minimal distortion of signal due to electrode positioning, and disadvantages that the signal strength is reduced due to distance. More number of signals are recorded and averaged. Then brainstem auditory evoked potential. These are another methods in which some signal is given in the auditory canal and some clicks are produced, and it is recorded by the by the brain. And there are this procedure we usually employ to test the functional integrity of the auditory pathways. It is very important to use this for posterior fossa surgery, which may impair the gearing or damage structures in the midbrains, pons, and medulla. Visual evoked potential, again, some flashlight is given on the retina, and it will produce the signals in the brain, and which will be recorded over occipital, parietal, and central scalp. So they test the integrity of the visual pathway. Previously, it was the auditory evoked potential which were testing the auditory pathway. Visual evoked potential, they are testing the visual pathways. This is a least commonly used technique nowadays. Motor evoked potential, this is the one of the commonest events which commonest modalities which we use. Nerves to be tested are stimulated, and response over the innervated muscle is recorded. The electrodes can be surface electrodes or needle electrodes placed directly on the innervated muscle. Small metallic electrodes are placed into the scalp over the motor cortex, and the response may be measured at the spinal column, peripheral cranial nerves, or innervated muscle. Nerve stimulation is at the site proximal to the operative area, and measurement of response is at innervated muscle. It can alert you the possible damage. Very commonly used. Nowadays, we use it for spine surgery, where surgeon is operating on the nerve roots and he doesn't want to damage the nerve roots. So motor evoked potentials are recorded, but for our purposes, the difference is that we have to avoid using the muscle relaxant. So we use a type of anesthesia which is known as total intravenous anesthesia, in which patient is anesthetized but still free from the effect of the muscle relaxant. So the patient even can be woken up during the surgery and asked to move the foot or dorsiflex or plantar flexor tube because foot, because patient is not under anesthesia, he is under lighter plane. But motor evoked potential will measure and forewarn the surgeon and anesthesiologist that the nerve root may be damaged or is about to be damaged. Electromyography tests the ability of nerve to produce a response in innervated muscle, and there are several responses which can be recorded. Electromyography alone is now not used. It is used in conjunction with the other nerve pathways, which is called nerve conduction velocity as well as EMG. So they tell you about the neuromuscular junction and state of the nerve. This I use more commonly because I have been dealing with the nerve injuries, acupuncture, and their peripheral regeneration after Bell's palsy, and these where it is very commonly used. NCV as well as EMG, both are used to predict the recovery as well as to establish the diagnosis of lower motor neuron palsy. Basically, what we measure the latency, we measure the amplitude. Latency, as you know, is the time from actual stimulus to the onset of response. An increase in the latency by more than 10% is considered significant at most centers. Similarly, amplitude, if the height of the wave, it is the height of the wave response, a decrease in amplitude by more than 50% means it is significant. Both these parameters, they help the anesthesiologists and surgeons to identify intraoperative neurological damage. So this is in short. I have made it very, very simple just for your benefit that that it is easily understood. Thank you for the patient hearing, and if you want any questions, I hope there would be.
Thank you, Dr. Anup, for your impressive presentation. I have a question.
Yeah.
What are the intraoperative sequels of mismanagement of depth of anesthesia? Like if there was a case of mismanagement, then what are the
that is what I was
results?
about. The patient may be relatively light during anesthesia. It is kept deliberately under lighter plane of anesthesia when we have a patient with multiple comorbidities and we do not want to take the patient very deep. In such cases, the main problem occurs during post-operative post-operative cognitive dysfunction. Patient may have some hallucinations. Patient may have some irritation, may have lack of sleep. These are the things which are seen in post-operative. During intraoperative period, what happens that the patient is kept under lighter plane, then which leads to increase in blood pressure, increase in the pulse rate, and patient may have some kind of ischemic changes to the heart, which we do not want during the surgery. So these are the complications which occur during surgery and in post-operative period. That is why it is very important to measure the depth of anesthesia, either by MAC or by one of these monitors.
Okay. So is overdose of anesthesia linked with cardiac arrest? Is this an unusual case or this happens?
See, we never take the patient to death that depth that patient has a cardiac depression or has a decrease in blood pressure to that level that ischemia occurs. We never take that patient to that level because most of the drugs available now are very, very good. They are not like the previous ones. See, previous ones, people used to use total one agent like ether, like cyclopropane, to provide all the three components of anesthesia, like muscle relaxant, also reflex separation also, and anesthesia also. Now we have different type of agents for different functions. For induction, we have different. For maintenance, we have muscle relaxant. For reversal, we have other agents like neostigmines and atropine. So we have devised different agents for different purposes. And combination of all this is known as a balanced anesthesia. That is why anesthesia has become so safe. But there are few conditions where patients have multiple problems. Like we are taking the patient for liver hepatic replacement, where a part of liver is given by the donor and put. Such patients already have lot of abnormalities. We cannot keep them at very high, very deep level of anesthesia. So we keep them with minimal level of anesthesia, but the recovery is facilitated. And if such thing occurs, nowadays the thing that if there is a prolonged surgery, now what we do? We don't let the patient recover on table. What we do? We put them on tube and send them to ICU for elective ventilation. And once the patient stabilizes, all these functions, blood gases, the electrolytes, the acid-base balance becomes okay, then only we recover the patient willfully. Okay, but this is for the patients who are under anesthesia for smaller period of time, say 3 hours, 4 hours, and their recovery is expected on table, where this monitoring becomes important. Okay.
Actually, I asked this because one of my batchmates from the PhD days, she went under a gallbladder, gallbladder removal. She was a polio patient, a girl of 35-year age, and after the completion of the operation, she had a cardiac arrest and she died. So obviously, we were not expecting it was a small operation, and we thought that it is because of the mismanagement or properly not managing the depth of anesthesia. So that's why.
No, it's not. There are other problems also.
And it happened in age. So
it may be because of the adverse reaction or some kind of hypoxia which has taken place during surgery. And gallbladder is a very tricky surgery. See, gallbladder also has a cardiac link. So it was known as Saint's triad before. So you have to be very, very careful monitoring the blood pressure because it is the upper GIT and the reflexes are very, very strong in those areas because the celiac plexus lies just close to it. So what we do most of the times now, if we are doing an open gallbladder surgery, we block those reflexes with the use of epidural anesthesia and then give a general anesthesia along with it. And nowadays, most of these procedures are taking place by laparoscopic means, which is a shorter procedure and in which it is minimal access. The major incisions are not given and recovery is on table. So the advancements have been there. I don't know what could have been the problem at that time. Maybe it may be a adverse reaction to the drug. That is more common. Normally,
doctors also said that this was rarest of the rare kind of a case, that something like that happened.
Yes, the mortality due to anesthesia has become much less after 1980, and that is because of the intense monitoring which we have in most of the hospitals, and we now have the advanced workstations which we work with, and we have monitoring like SpO2, ECG, NIBP, which is continuously there with EtCO2 monitoring, end-tidal carbon dioxide, which is monitored all the time. And in between, we take the blood gases at that time to see the status of the electrolytes and this thing. So it is highly unlikely in these times. But yes, I agree that there is a lot of discrepancy between the major hospitals and periphery in our states. I have been to number of centers. I have visited maybe more than 50 centers by the government, and they have very few anesthetists and very few age-old machines which they are using. But now, not in Delhi. Delhi, everybody has the minimum monitoring standards, which is there by ISA, and everybody has to follow that. So with the monitoring, these things have become much better. So depth of anesthesia is another. This monitor is one of the new things in our armamentarium. It gives us a number so that we know that we have to keep the patient between this figure, then patient is adequately anesthetized and will wake up very nicely.
Professor Gogia, thank you very much indeed for this insightful conversation on this important topic. We'll now take a short tea break, and I request all participants to rejoin by 3:30 p.m. In what would be a wonderful conversation by Professor Pallab Bhattacharya when we meet. Thank you.
Thank you so much.
Welcome everyone to this post-tea session. Professor Pallab Bhattacharya that can improve neurological deficit.
I think there is a technical glitch. Imran, can you repeat?
And
yeah, your voice is cracking. Just
Is it audible
for
to
a
you?
second. Pallab, just wait for the.
Yeah, no problem. No problem. You can hear me, right?
Yeah, you are audible. Yes.
Okay, thank you.
Imran is going to introduce you. Then
sorry, Professor Bhattacharya, please. Yeah, you may please continue. Sorry.
Yeah, so I'm going to share my screen now, and just a minute. Can you see my screen?
Yes, it's visible.
Yeah, so I will put off my videos to just facilitate the internet stuff here. So a very good afternoon, everyone. And thank you, Professor Suri, for giving me the opportunity to talk to you people and to present my research before you all. Thank you, IBRO, for the funding that they have provided. So today I'm going to talk on research that my team is pursuing at National Institute of Pharmaceutical Education and Research, which is an institute of national importance under Government of India. So my lab basically works on translational aspect of stroke neuroprotection, where we are intervening stroke by stem cell therapy. To start with, first of all, my disclosures. So DST, ICMR, and all the fund bodies, those who have modestly funded me during my research. So first of all, for all the junior colleagues and students, what actually stroke is. So stroke occurs when you have an obstruction within a blood vessel supplying blood to the brain. And it is one of the leading causes of death worldwide. And most of the patients who have a stroke, they do have ischemic stroke. If you look into the categories, so stroke has been categorized into 5 types by American Heart Association. It can be ischemic, it can be hemorrhagic, it can be a transient ischemic attack, and 2 new categories have come, one being the brainstem stroke, another is the cryptogenic stroke. Cryptogenic stroke is the stroke which you do not know the reason why the stroke has happened. So if you talk about the ischemic stroke, it can be caused due to thrombosis, systemic hyperperfusion, cerebral venous sinus thrombosis, or embolism. Where hemorrhagic stroke can be caused due to brain trauma, tumors, arteriovenous malformations, or aneurysms. So if you look into the percentage of stroke, approximately 85% of all strokes are ischemic in nature, while rest comes under the percentage that is beyond 85%. So it becomes imperative for us to learn more, to delve more deeper into the reasons, causes, the mechanisms involved, how you can render neuroprotection in stroke in the case of ischemic stroke. Now, if you look into the stroke facts, in India, between 145 to 154 per 1 lakh individuals a year have a stroke. On an average, a stroke occurs every 40 seconds, and every 4 minutes, someone dies of stroke. Half of all strokes in India can be attributed due to hypertension. And 1/4 of all strokes in India occur in people who already had a stroke before. It can be a lacunar stroke or it can be a mini stroke, but they might have stroke before. As this requires a more advanced clinical scenario, most advanced clinical settings to treat. So it becomes very much tough for the patients from the rural areas to reach advanced clinical settings to get it treated. So it becomes one of the major causes of death. You can say number 1 cause of death when you talk about the rural population. Now, if you look into the risk factors, and if you categorize between the elders, approximately 60% of strokes occur in the age group of 74 to 84, and 80% in elderly people who are above 85 years of age. And there also being a gender disparity, if you look into the percentage of the stroke occurring in male and females. So 52% of stroke occurs in male who are coming in the age group between 65 and 84, while 48% in females. But if you talk about more elderly people, those who are above 85 years of age, females are more vulnerable. Means they have 63% of stroke in comparison to males who only have 37%. Now, major thing that how you can recognize the stroke. If any one of your relative, if any one of your known person is having stroke, how you can recognize it? Because it is one of the factors that ask you to take the patient to the hospital. So first you will find that the person is losing balance. He has a headache, dizziness. He may have a sudden loss of vision in either of the eyes or both the eyes. He may have a drooping of the face. He may have an arm weakness. His voices are slurred, he's facing difficulty in speech. And that is the point of time where you should not waste time and you take the patient directly to the hospital. And that what is the most important, the time is brain. Now, presently in the Indian clinical aspect or the clinical aspect of the world, the two major therapies are FDA approved. One is the tissue plasminogen activator that when you take the patient to the clinical setting, they give a TPF, and that has a limitation that it has to be given within a limited time window of 3 to 4.5 hours. And if you take the patient to advanced clinical settings, they do a mechanical thrombectomy where they pull out clot by using a catheter, and they insert the catheter from femoral artery, take it to the brain and pull down the clots. But this mechanical thrombectomy is not so rampant in the Indian clinical aspect because it requires a high-end clinical settings, and all the hospitals and all are not very much equipped with this kind of facility. Well, what to do? You have limited treatment options. The time window is very important, and you cannot recoup the brain neurons which are dead. So what to do? Now, the strategy is to protect those neurons which are surviving or which are in a mode where they are not either dead or they are not functioning well. So if you talk in the terms of biological system, there are a few strategies in neuroregeneration. Either you replace like cells for the like cells, or you stimulate the endogenous repair mechanism that in our brain you have a subventricular zone neurogenesis. And if you just promote SVZ neurogenesis, you can compensate, you can promote the endogenous repair mechanism. You can halt or you can prevent the ongoing injury, or you can either modify the toxic microenvironment. Now, where comes the concept of stem cell therapy? So stem cells from past have been reported to have the ability to repair, replace, and regenerate. There are many types of stem cells. It can be neural stem cells, embryonic stem cells, mesenchymal stem cells, hematopoietic stem cells, and induced pluripotent stem cells. For our research, we usually take mesenchymal stem cells. The reason for that is being that these cells have a high potential for stroke treatment. There are no ethical concerns as they are from non-embryonic sources. They are immunoprivileged, or you can say they lack major histocompatibility 2 and less major histocompatibility 1 complex. They are easily produced. When you talk about intervening stroke, there can be multiple routes of stem cell delivery in that condition, or even in any pathology. If you talk about neurological disorders, there can be multiple routes how can deliver stem cells. It can be intravenous, intraparenchymal, intracerebroventricular, and intraarterial. So we basically in lab, we have chosen the intraarterial route because these are minimally invasive and this evades the systemic effects, and this has a high potential for clinical translation. What I mean of a high potential for clinical translation, because most of the endovascular therapy, if you're talking about the mechanical thrombectomy, they take the endovascular route for its accomplishment. So if you anyhow access that artery, it can be benefit, it can come as a benefit, and it can be superior route of administration than other routes available. I will talk more detail about the why we have chosen this intraarterial route. So if you talk about the history of stem cell therapy, it hasn't come a single day. The first successful allogenic stem cell therapy was carried out in 1957 by Donald Thomas. And he won the Nobel Prize for the same in 1990, where the bone marrow cells were transplanted from the identical twin of a patient suffering from acute leukemia, who was treated with supralethal whole body radiation. Although it was a failure, and it was ultimately failed to cure the leukemia, but it was the first example where the stem cell therapy came into the picture. The next study was done by Robert from the University of Minnesota in 1968, where he transplanted the bone marrow from one of the siblings to the other, and he was able to cure a child who was suffering from SCID, And he reconstituted his lymphoid and hematopoietic functions, which were revived by the transplantation. So you can see directly it was one of the success story of stem cell therapy, and that it initiated from this study. And later, in the University of Glasgow, Queen Elizabeth University, a single intracerebral dose of neural stem cells up to 20 million induced no adverse events and were associated with improved neurological function, and that what we call as the stem cell in stroke PCS trial. So this was a history where stem cell therapy has shown benefits in the case of stroke and in other pathologies as well. Now, as I told you before, that why intraarterial, why not intravenous? So if you talk about certain studies who have done that, this intravenous intraarterial route of delivery has some limitations as well as some positive aspects. So if you push the cells intravenously, then there is always a chance that this does not circumvent the first pass. It accumulates near the spleen and liver region, and it may not reach to the brain foci where you have the insight. But if you talk about the intraarterial delivery, if you take the carotid and you push the cells directly to the brain, the cells can directly reach to the stroke foci, and you have the maximum number of cells at the stroke region. So it was Michael Chalk from University of Michigan who conducted one study where he saw that approximately 4% of the cells which enter through intravenous route reaches to the brain and survive, while 20% of cells enter the brain and survive in the case of intraarterial delivery. Another study from Rafael Guzman, who was a subordinate of Gary Steinberg from Stanford, he conducted one experiment where he saw that the 74 cells per mm square in infarct lesion was seen when the cells were delivered intravenously, while 1300 cells per mm square of infarct, the cells when it were delivered intraarterially. So it was anyhow proved by other studies that intravenous proved to be less efficacious, where intraarterial delivery was more efficacious. And that forms the basis that it has a high potential for clinical translation. Now, it was Michael Chalk again who did some preliminary experiments where he took 3 groups of intraarterial, intracerebroventricular, and intravenous, and he injected cells which were packed with nanoparticles. These were all SPIO magnetic nanoparticles, and he pushed the cells by 3 routes. Astonishingly found that when the cells were delivered by IA route, within 4 hours, the cells were there sitting in the brain. But if you talk about the IC and IV, even if up to 4 days, you only see a few punctate signals coming out from the stroke foci. So this example or this experiment anyhow gave a boost how the intraarterial can transform into a clinical perspective for stroke treatment. Our lab in the US, they also did a biodistribution cells where they injected cells, IA and IV, and they measured the proton flux coming out. And you can see that in the case of IV, most of the protein flux are coming out from the splenic and liver region. In the IA, you have the signals coming out from the brain. And up to day 7, you find the signals, means the cells are over there. Now, before going to the clinics, there were certain questions that we needed to address. Firstly, you should have a model which should be clinically relevant, and that study being conducted in that model should be able to translate. Second, there were general concerns of the blood-brain barrier, although we know that whenever you have a stroke, you have a compromised blood-brain barrier, but it is very differential. You do not know when the blood-brain barrier opens and when the blood-brain barrier closes. And it is very different for different people and different time points. Now, when you talk about intraarterial, you are pushing cells to the brain and you're pushing the cells directly into the blood circulation. Now, the cells are obviously bigger than the blood corpuscles. Now, the question arises, do they really compromise the circulation? And if they compromise the circulation, they even cause a microembolic stroke. You are going to treat stroke and you are coming out with a lacunar stroke. That is also not acceptable. Now, when you push the cells, how transplanted cells effectively reach to the site of injury? Do they survive or phagocytose in the brain? How the blood microenvironment or the brain microenvironment behaves with the cells and vice versa? How the toxic effects? What is the mechanism of neuroprotection? There are many questions before us which need to be addressed when you're thinking of taking your research from bench to bedside. So we had a very good model. Later on, I will talk about the large animal model as well. So we have most accepted MCO model. The MCO model is a middle cerebral artery occlusion, where we push the filament from the external carotid and push it through the internal carotid towards the middle cerebral artery. And we have a descent stroke. You get descent infarct. This is a TTC stain, a nasal stain, where you see that you have a descent stroke in the rat brain. And from that, we also conduct a real-time blood Doppler and brain oxygenation, where we see that how there is a difference in the cerebral blood flow, whether the cerebral blood flow is coming down when you're occluding the MCA or not, and how the brain oxygen content is at that point of time where you are making a stroke model. And we also we ventilate the animal, we cannulate the animal in the femoral artery and femoral vein. We intermittently take the blood and we just go for blood gas analysis, and we try to keep all the blood gas in the normal limit. Apart from that, blood glucose has always been kept constant, or it should not exceed, or if it exceeds, we just exclude that animal. So when you have a stroke, you have the neurodeficit score where the rat shows the circling movement. So we should remember that if it is a left side stroke, the rat should have effect on the right side. If you have a right side stroke, the rat should have an effect on the left side. So we have a staining, we fix the biological parameters, we see the blood oxygenation, we see the cerebral blood flow, we score the animal, and after that we have a model which mimics the actual focal ischemic model that occurs in the clinic. Now, the first question arises that what are the efficacy, what are the preclinical safety and efficacy of our study? So we started to optimize the time point and the number of cells that needs to be pushed when you are going to treat cells. So we didn't know the value of the number of cells that needs to be pushed and can show benefits. So we started with 1 million cells and we de-escalated the dose. And we found that 100,000 cells didn't worsen the cerebral blood flow. And this can have a beneficial effect when you talk in the sense of neuroscore. And the neuroscore was taken all together for 28 days and the animals were survived. And the interesting question that came before us, when you inject cell at 1 hour, there is no benefit or there is a less benefit. But when you inject same number of cells at 24 hours, you see a remarkable benefit. Now you have the same model, you have the same number of cells, you have the same route of delivery. Why delivery at 1 hour is not efficacious and why delivery at 24 hours was efficacious was a big question before us. So we started growing into the literature and we found that there is a mechanism of STF1-CXCR4, where when you have a stroke, there is an upregulation of stromal-derived factor 1, and that is a chemoattractant where the MSCs express CXCR4 receptor, and they directly goes and attach to that receptor, and they usually move towards the stroke foci. But we had to validate with the experiment. So we conducted STF1 study, and astonishingly we found STF1 was more at 6 hours and it was continuously up to 24 hours. But at 1 hour, there was not much significant amount of STF1 that could attract these cells, that could enhance the homing of the cells, and the cells could reach the stroke foci. Now, when we were communicating this paper, the reviewer came to us that, okay, you are talking that at 6 hours, the SDF1 is upregulated. What about the blood-brain barrier? So we argued that, okay, whenever you have a stroke, you have a compromised blood-brain barrier. But again, the question comes that it is very differential. It may be with one animal, it may not be with the other animal. The blood-brain barrier opening at 6 hours may be with some person, may not be with some person. So in that case, we conducted one experiment where we injected the Evans blue dye and we observed the envisagement of the Evans blue at 6 hours. So now we were confirmed that the time point that we are taking to push the cells and the time it has a blood-brain barrier opening, and we have SDF1 maximum expressed, so the cells will reach to the stroke foci. Now, these two factors were not only the factors. We were also concerned with the stem cells. Now, when we are taking the 100,000 cells, we made sure that we do not go for a higher passage number. Why passage is important? Because when you go for a higher passage number, the cells have a genomic instability. And when they have a genomic instability, they have the tendency to differentiate. We don't want these cells to differentiate at the brain, because you do not know how this differentiation is going on. You are going to treat stroke and you land up with glioma. We don't want that. So we want that stem cells should go, they should give some of the factors, they should just go off, leave the brain and go out, and the neuroprotection time window should extend. Now, time point was fixed, dose was fixed. Now, the question was that when you are pushing the cells, whether you are getting resistance in pushing those cells because you are facing the robust circulation. Yes. So in that case, when you try to push cells harder, in that case, you have a chance to get a microembolic stroke because the rat brain capillaries are very small. In that case, the duration was also optimized, and this 100,000 cells were pushed with a PE10 catheter, which was inserted in the intracarotid, and it took 10 minutes to push these 100,000 cells, which was dissolved in PBS. Now, the question was that if it is a PBS and if it is the MSC is dissolved in PBS, the infusion should not vary. In that case, I will come back to the other slides. I will just show one of the feed. So this is an experiment where we took PBS and where we took MSCs. And with this 10 minutes pushing of the cells and PBS, there was no much deviation in the laser Doppler flowmetry. So in that case, we have tried to figure out that even if it is a PBS, which can easily move into the brain circulation, the MSC is not going to harm or rather is going to produce any microembolic stroke. Coming back to previous slides. Now, as I mentioned that it was the SDF1 CXCR4 axis, now it needs to be confirmed when you have to treat the animals with the CXCR4 antagonist. So this was a brain biopsy study where we found the relative abundance of transplanted MSCs. And if you see those groups which has a CXCR4 antagonist, they didn't have much number of cells in the cortex, either in striatum. So this was an experiment that confirmed that the brain cells were pushed to the brain foci, and there were the CSDF1 CXCR4 axis are playing a role where the stem cells are directly going to the stroke foci and sitting at their site. Now, as I mentioned, we also characterized the stem cells. We did the FACS. We also faxed down cells and we took considerable diameter of cells. But the main important thing that we tried to look into that it should have the CD45 negative and it should have CD105 positive. Now, for all my young friends who are listening to me, the CD45 is a transmembrane tyrosine phosphatase. The expression of the CD45 determines the multilineage differentiation capacity. And if you talk about CD105, CD105 positive are less prone to differentiation. So this is what we want, because if you're trying to take your study to the clinics, you need not to work more on the characterization of stem cells. You only need to see whether this is going to help you out and these cells are compatible to the body or not. Now, in past, we also have the hint from the studies that the cells anyhow are giving neuroprotection by the help of neurotrophic factors. And the brain-derived neurotrophic factors, BDNF and stroke, had a very, very intricate relationship. So what is BDNF? BDNF is a member of a neurotrophic factor, and it is one of the most powerful neuroprotective agents. In animal model of MCO or the global cerebral ischemia, it has been demonstrated that intraventricular administration of BDNF protects the brain from ischemic damage. But the question arises, if it is only the BDNF, you can only give the BDNF to the brain. But the challenge is that the BDNF is unable to cross the blood-brain barrier. So we also had some of the protein expression studies of BDNF and its receptor tract B, and we saw that IMSC treatment after stroke increases BDNF and its receptor proteins in the penumbra. So our preliminary results even gave a hint that how BDNF is helpful in neuroprotection. Now, tracking of MSCs and survival. Now the question arises when you push the cells inside the brain, how you track the cells. When the cells are in the brain, you cannot see, you need a specific imaging system. So we had two confirmatory experiments. One was a microscopy and other was the fluorescent in situ hybridization. What in fish we have done, we took GFP positive male animals, we harvested the bone marrow, we took out the cells and we injected in the female animals. Now female has estrogen, it can have a neuroprotection. So we over-ectomized the animal and injected those cells to the female animals. Now if you see, due to the XY disparity, you can find the signals coming out from the fish. And if you talk about this microscopy, I will give you more clear picture. This is the brain structure, which was before the infusion. These are the capillaries. You have the infarcts. And this, the second one, if you see, you have the GFP positive MSCs all around. You can see the small glowing GFP cells. So now we were very sure that our cells which were pushed into the brain, they are sitting in the brain infarct and they are there. Okay. Now, we also had a 2-photon imaging system and we wanted to see for how many days the cells were sitting in the brain. So we found that the cells, the number of cells started decreasing, and anyhow, after 7 days, you only have a few in the brain. So this gave us a hint, cells are not sitting in the brain throughout. They are there for over a period of time. And after that, they are either phagocytosed or they are even, we still do not know the fate. But one thing we know, that the time window of neuroprotection was extended, and you get a neuroprotection in extended time window. Now, a big question before us, that when you're pushing the cells to the blood, to the arteries, the artery has blood. So how these cells behave to the blood? Now, you already have a stroke, and when you push MSCs and when it comes in contact with the blood, do they trigger an inflammatory response? Do the cells lose their viability? These were questions. Is there any toxicity in the animal or not? So we went for some hemocompatibility studies and we found there were no adverse reaction in either of the groups which had a share with IAPBS or MSCs. We also went for hemocompatibility studies where we also checked for different blood parameters, and above all, we also tested for D-dimers, MPT, APTT, and we found that all were in the normal ranges. We also went for toxicology study where we went for LFT, KFT, and these are the different sections of kidney, brain, lung, liver, spleen, stomach, and heart, where we checked that there were no adverse effects. Well, it was very clear to us that the stem cells are not causing any harm to the body, and this can be taken into the clinics. Now, when we talk about mitochondria is very crucial in stroke. Now, for all my young friends, what actually happens, I just give an overview. So when you have a stroke, there is an increase in the intracellular calcium levels. And this calcium then moves into the mitochondria by the calcium uniporter, which are present on the mitochondrial membranes. Once this calcium concentration inside the mitochondria increases, it leads to activation of several detrimental processes, one of which is the formation of mitochondrial permeability transition pore that we call as MPTP. And this MPTP formation itself is a trigger of the leakage, and it leads to leakage of essential ions from the mitochondria, destabilizing the mitochondria. high calcium concentration interferes with the ETC, which leads to generation of ROS, which is a high concentration overwhelms the antioxidant system within the mitochondria. Now, the high ROS also disrupts the mitochondrial membrane, leading to release of cytochrome C from within the inner mitochondrial space. Now, this cytochrome C release pushes the cell towards apoptosis by activation of different caspases. And the mitochondrial membrane disruption also leads to leakage of ROS into the cytoplasm, eventually leading to the damage of the lipids, proteins, and nucleic acids. So there are a lot of things, and mitochondria being one of the important cell organelle in stroke, and you have ATP depletion when you have a stroke. So the roles of mitochondria is very crucial. Now, what we try to hypothesize that whenever you push the cells to the brain, these cells might come in contact with the neurons of the brain, which are surviving. Yeah, I'm not talking about the dead neurons, because those neurons which are dead, you cannot do anything with them. We have to salvage, we have to save those neurons which are in hypoxic environment, or they have an altered function or which are dysfunctional. We try to help or we try to rescue those neurons. So we hypothesize that these cells, when they come in contact with these surviving neurons, they may form some tunneling nanotubes between them. What are tunneling nanotubes? These tunneling nanotubes are small tubes, which each and every cell make within each other to communicate. They communicate by donating cell organelles. They communicate by donating some of the factors. There are many things that can be communicated between two cells. So we thought that when we are pushing these MSCs, they might have some of the donation to the hypoxic neurons, and that too in the form of mitochondria. It was a hypothesis. We didn't know what is going to happen. So for that, we first designed one in vitro experiment to start, because we didn't know it is going to happen or not. So we cultured PC12, we kept it in the hypoxia chamber, and we co-cultured it with MSCs. And there we started looking into whether there's any formation of tunneling nanotubes there or not. But to our astonishment, we found we stained differentially. The MSCs were stained with mitochondria, and the PC12 cells that were cultured were stained with Vidjum agglutinin. And to our astonishment, we found the long thread-like tunneling nanotubes formed. These are some of the images which clearly show how the big cells are the MSCs and the smalls are the PC12, and how these nanotubes are being formed. Now, let me show you one of the 3D videos where you see that how MSCs and PC12 cells are communicating with each other. If you see the second live imaging, if you see the cells, the green cells, these are MSCs, and the red one are PC12. See how this PC12 and MSCs are coming together, and they are communicating. They are nanotube formation. Okay. So now in vitro, we were there that, okay, the nanotubes are being formed. Now we just started to see whether the mitochondrial transfer has occurred or not. So we wanted to see that when you culture this hypoxic PC12 cells with MSCs, is there any role of mitochondria from MSCs for its survival or not? So we took some of the studies where normoxic PC12 were cultured with MSCs and hypoxic PC12 were cultured with MSCs. And if you see the mitochondrial signals, those PC12 which were hypoxic and the culture, they have the maximum mitochondrial signals. Even we had the percent viability. If you see the percent viability, those of that PC12 normoxic and MSCs were equivalent to that of PC12 normoxic. And when you talk about PC12 hypoxic plus MSCs, it was better than PC12 hypoxic in singlet. So this was some of the hint, which was not sufficient, but it was a hint towards us that, okay, there are role of mitochondria and they are helping in surviving of the cells. We again went for the ATP consumption study in the hypoxic PC12 cell line. What we did, this was done by Max, and the PC12 normoxic cell and PC12 hypoxic cell, we had analysis of the relative ATP quantity. We also co-cultured PC12 normoxic, co-cultured PC12 hypoxic, and co-cultured MSCs and co-cultured MSCs, both normoxic and hypoxic. And we found that, okay, these cells, the energetic of cells are really changed, and there might be the movement of mitochondria towards the PCT cells. Now, when we checked the various gene expressions in the following co-culture of MSC with hypoxic P12 cell, and to our astonishment, the SIRT1 came to be one of the major players. And now we tried to figure out how SIRT1 is anyhow involved in the complete process. Now, before moving to the SIRT1, we should have a preliminary information about SIRT1. Certainly, these are NAD-dependent deacyl having a diverse role in energy metabolism, modulation, cell survival, transcription regulation, inflammation, circadian rhythm regulation, and DNA repair. SIRT1 dysregulation is said to play a key role in the pathogenesis of various neurological disorders. SIRT1 has been shown to be neuroprotective against cerebral ischemia reperfusion injury, and it has also shown to play or regulate the immune response by modulating the NF-kappa beta. Now, targeting SIRT1 dependent pathway paves a way for developing novel therapeutic strategies for cerebral ischemia. Now, we also have introduced the concept of inflammasome. What are inflammasomes? Inflammasomes are multi-protein receptor-like structure involved in the initiation of inflammatory cascades. Major components of inflammasomes are the ASC, caspase-1 and ALP, and these crucially plays important role in the progression of inflammation and apoptosis. Now, we hypothesize that whenever you have IMSCs, it will upregulate SIRT1, which in turn will upregulate BDNF, and this can downregulate, or this can halt the inflammatory mediators, which can reduce in apoptosis and render neuroprotection. For that, we designed a study where we had the preclinical model, we had ischemia, reperfusion, and then we sacrificed the animal and we went for different molecular studies to confirm the mechanism. So here what we have, apart from that, we also have an inhibitor group over here, which is the SIRT1 inhibitor, that what we call as EX527. This EX527 actually is a selective inhibitor of SIRT1, and its effects are very prominent when you try to elucidate the molecular mechanism. Now, this, if you see the TTCs, the post-IMSS treatment, significantly reduction in the neurological deficit were seen. Can you hear me? Yeah. Now, if we have different groups, now this group also had the inhibitors and everything. So if you see, the post-IMSC treatment significantly reduced the neurological deficit as well. When this is the neurological deficit score, you can see the graph at the right side, that while the neurological deficit were higher in the group which has stroke plus inhibitor plus IMSC animals, and consistent with the neurodeficit score, you can see the higher infarct size, which was seen in the stroke plus inhibitor, and stroke plus inhibitor plus IMSC group. But if you see, there was no significant difference between these groups. There was a significant reduction in infarct size when you talk about stroke plus IMSC group as compared to stroke group in singlet. Now, if you talk about the neuronal viability, you can see that intact cells were observed in the sham animals. If you see the morphology, there was aberrant changes in the morphology along with nuclear shrinkage. When you talk about the group stroke plus inhibitor, and stroke plus inhibitor plus IMSCs. So a higher reduction in the Nissl stain was seen in the stroke plus inhibitor, and stroke plus inhibitor plus IMSCs groups as compared to the sham. If you see the stain, the higher Nissl stain was observed in the IMSC treated groups. And there was a reduction in the average neuronal density and neuronal length in stroke plus inhibitor plus IMSCs group as compared to sham and stroke plus IMSCs group. as you know, when you have a stroke, you have a motor coordination deficit. In relation to that, we had a rotarod and grip strength study. So what is a rotarod? The retention time on the rod of the animals is being seen. And if you see, the retention time on the rotarod was significantly decreased in the case of stroke plus inhibitor, and stroke plus inhibitor plus IMSCs as compared to sham in 5 and 10 RPM. A decrease was also observed at 20 RPM. However, the decrease was not significant as compared to sham. Now, following IMSC administration, there was significant increase in the latency time that was found in the stroke plus IMSC group at 5 and 10 RPM. And at 20 RPM, there was no much difference seen as found with the different groups. The same with the case with the grip strength. In the stroke, it was significantly decreased as compared to the sham group. And the IMSC treatment group significantly had improvement in the grip strength, and as observed to the stroke group. And unfortunately, no improvement was seen in the grip strength of the animals which had the stroke plus inhibitor and stroke plus inhibitor plus IMSCs. So here are some of the mRNA expression studies of SIRT1, BDNF, NF-kappa beta, NLRP, NLRP3. So if you can see, the levels of SIRT1 and BDNF mRNA were significantly reduced in stroke plus inhibitor plus IMSCs. But when you treat the animals with MSCs, this MSCs treatment upregulated the mRNA expression of SIRT, BDNF, and apart from that, conversely, the mRNA level of NF-kappa beta, NLRP1, NLRP3 were significantly elevated in the stroke plus inhibitor, and stroke plus inhibitor plus IMSCs group, while the MSCs treatment reduced the mRNA expression of these markers in stroke plus IMSCs group. This is the array of the protein expression study, where the protein expression of SIRT1, BDNF, caspase, TNF-alpha, NF-kappa beta, NLRP1, and NLRP3 were seen. And the most important thing that you have to note over here, that an increase in the expression of SIRT1 and BDNF and TRAC-B protein were observed following the MSCs treatment. And in the same group, MSC treatment reduced the expression of TNF-alpha, NF-kappa beta, inflammasome, that is NLRP1 and NLRP3, and the apoptotic marker cytochrome C and caspase 3. So this was a good hint to us that, okay, these MSCs have a beneficial role when you take it for the treatment of stroke. So these are some of the inflammatory mediators, TNF-alpha, IL-6, and IL-1 beta. So if you see, the levels of inflammatory mediators, TNF-alpha, IL-6, IL-beta were significantly elevated in the stroke group as compared to the sham. But there was no significant difference observing the expression of these mediators in the stroke plus and stroke plus inhibitor group. But when you gave IMSCs, the treatment significantly reduced the expression of these mediators, and the levels were found to be elevated in the stroke plus inhibitor plus IMSCs group. So this was a good hint towards the beneficial role of stem cells in the case of ischemic stroke. We also had the tunnel, and there was a robust expression of tunnel positive cells and in the stroke group as compared to sham. And the similar results were observed in stroke plus inhibitor and stroke plus inhibitor plus IMSCs group. The number of tunnel positive cells were significantly reduced in the stroke plus IMSCs treated group. So we counter-confirmed all of them, the markers with the immunohistochemistry. And this is the immunofluorescence imaging of the BDNF. The same trend was seen. This is the immunofluorescence image of NF-kappa beta, the same trend as it was observed. This is the immunofluorescence image of NLRP1. It was absolutely concurrent with that of the protein expression study that we got. This is the NLRP3. And this is the tunnel, which has the same results that were obtained before. Now, do we have time? Organizer, do I have time for 15 minutes?
Yes, you can take some time.
Yeah. Now, there were certain questions coming up in literature that you are talking about SIRT1, why not SIRT3? Because some of the papers have reported that SIRT3 also has some of the role of neuroprotection in the case of stroke. Now, we did a pilot experiment for that. we checked the SIRT3 expression in sham, stroke, and stroke plus IMSCs. Now, the most discouraging result was that when you talk about sham and stroke itself, there was no significant difference in the expression of SIRT3. It was concurrent when we went for immunofluorescence study. And although stroke plus IMSCs has increased SIRT3 expression, but when we didn't find any difference in sham and stroke itself, it was hard for us to carry SIRT3, although a few papers have reported its beneficial role. So rather we have concluded from that SIRT1 and not SIRT3 is majorly involved in the IMSC-mediated neuroprotection, and that too in the case of mitochondria. Now, this was, again, if it is SIRT, then we should also have an activator-inhibitor study. So this was the effect of SIRT1, an inhibitor on the neurological deficits and infarct sites following IMSC treatment. So this is a TTC, and if you see, resveratrol is a SIRT activator, whereas EX527 is a SIRT inhibitor. So we compared both, injecting both in the groups, and tried to decipher whether IMSC is working as a SIRT activator or not. So we found that when you have stroke plus resveratrol and stroke plus resveratrol plus IMSCs, so in that case, the difference was not so distinctive. If you talk about stroke plus EX527 IMSCs. So in that case, IMSC group has more viability in comparison to both inhibitor as well as activator. We again moved for some of the studies where we wanted to see the neuronal viability. This is the Nissl staining, SIRT1 activator, SIRT1 inhibitor, and the study clearly mentions that IMSC group has more viability in comparison to both inhibitor as well as the activator. We had the area of protein expression study, and we also plot the TNT markers. And if you can see, the TNT markers were upregulated in the presence of IMSCs. Mitofusing markers increased, and mitofission markers were decreased in the IMSCs group. And if you look into the mitobiogenesis markers, these were also increased when they were treated with IMSCs. We did a PCR and we found the concurrent result as to that of Western blotting. Now, if we also went for the ISC of SIRT1, and we found that IMSCs increased SIRT1 activity, while the SIRT1 inhibitor inhibited the SIRT1 expression. We also went for ISC of ROT1. ROT1 is for the movement of mitochondria through the tunneling nanotubes. So IMSCs increase ROT1 expression, while ROT1 is inhibited in the SIRT1 inhibited group. We also had a ISC of RALA, which is a TNT marker, and IMSC increased RALA expression while RALA was inhibited in SIRT1 inhibitor group. We also had tunnel, and tunnel positive cells were decreased in IMSC's group. We also had mito-ROS. We use the mitosox dye, and this dye only binds to the mitochondrial ROS. And we have found that IMSC decrease the mito-ROS generation. We also had the mitopotential by JC1 dye. So how we do measure? JC, the red color is for the healthy mitochondria, which has a red fluorescence, while the green is the dysfunctional, which will give us the green fluorescence. And red and green ratio actually gives the actual value of the mitopotential, and we have seen the mitopotential increases in the IMSS group. We also had the different complex activities, and overall, the IMSS improves the complex activities of the mitochondria. We also checked with the ATP level, and we have seen that IMSS increases the ATP level in the treated groups. We also had the various biochemical parameters of GSH, SOD, nitrite, MDA, and in all the groups, the IMSCs were able to normalize the biochemical parameters that were aberrant in the cases of stroke. We also had the expression of various inflammatory mediators, and we have seen that IAMSs reduces the inflammatory mediators. Now we have a preliminary result. I have more studies there, but due to paucity of time, I cannot go deeper into that. So let us come to the canine model where we have validated the canine model. We have a large animal model where we put the coil at the M1 region and produce a stroke. You can see a decent infarct in the dogs. Now, we treated the animals. The dose optimization was 1 million cells of MSCs, and we treated them at 24 hours. And after 30 days, you can see the infarct area is being reduced. This is another set of animals which had a reduced infarct area. We had a comparative group where we have seen that at subacute, when you treat IMS subacutely, it does not exacerbate the stroke injury in dog. This was IV, and it was a comparative group with the IA, and you can see there's no such benefit when you see ex vivo MRI of a dog brain. Now, we also went for diffuser tensor imaging, and the diffuser tensor imaging clearly mentions about the growing fibers, which were seen after IMS therapy in case of canines. So that's all from these are my collaborators, my students, and thank you very much for your attention.
Thank you, Dr. Palak, for your interesting talk. There are some questions in the chat box, so I'm going to read.
Yeah.
First question is that why can't neural stem cells be used instead of MSC since they are more specific?
Okay, now when we talk about neural stem cells, we know that these neural stem cells are going to form neurons. Now, it is not only the formation of neurons, the neurons has to make effective connection with the persistent neurons. So if you just put up some neurons in the brain, you cannot say that these neurons are functional neurons or they are going to give an effect, because they may not make some functional connections with the persistent neurons. Now, you also do not have a control over the number of neurons which are produced. So you are trying to treat the patient with neural stem cells and you are coming out with a glioma that is also not desirable. So in the case of MSCs, these are safe, as I have mentioned, that they have, we try to control the differentiation of MSCs, and we only want that factors should work. And it's not the differentiated cell should work. The cell should do their job and go out of the brain. That's what is required. And that's what is clinically relevant as well.
Okay. I think the second question is linked to your answer. So someone has asked that, did you do any assessment of the differentiation of the fate of the transplanted MSCs? Even if,
sorry,
so did you do any assessment of the differentiation fate of the transplanted MSCs?
Yes, yes, we have studies. I haven't shown that data over here. We have studies, and I mentioned 1-2 photon microscopy image where we see that after 7 days the cells go out; they are not visible. So these transplanted MSCs, either they are phagocytosed. There are some studies which have shown that these cells are phagocytosed in the brain, and they can also go for apoptotic cell death. But the correct mechanism, whether they are phagocytosed, whether they are going for apoptosis, is still under investigation. But anyhow, the neuroprotective time window is there. The cells are working, and you can see in comparison to the stroke group, you have improvement in the functional outcome.
Okay, so I have a question.
Yes.
So in the end, these MSCs are helping by providing healthy mitochondria to the damaged cells. And after providing these mitochondria, they are probably being going into apoptosis or the type you mentioned. So what happens is, if the mitochondria is mutated, so it becomes a heteroplasmic state for the cell that is being repaired. So there is a damaged mitochondria already there, and there is a healthy mitochondria coming from the MSC. And these damaged ones are they are known to overpower the functional ones, like the dominant negative mutant states. So is this a permanent repair, or does it changes after some time? The damaged mitochondria, they take over the newly entered mitochondria. So what's the possibility actually?
No, the thing is that when I talk about the MSCs and when I talk about the donation of mitochondria from MSCs, I only mean that the cells are communicating and cells are giving their mitochondria. Now, when you talk about the mitochondrial deficit, when you talk about the mitochondrial genetics, no one knows what kind of mitochondrial genetic each and every person has. So now it is not a kind of a personalized medicine. We are trying to figure out a therapeutic aspect which can have a mass effect. Now, for the time being, if you are seeing that the cells are going to the brain, the MSCs are pushing the mitochondria towards the neuron, and the neurons which are in the hypoxic environment, which are dysfunctional, are functional. So that is the point where you are having the clinical relevance. Now, when you talk about, there are many things, even I can say that we can engineer MSCs, but this can better survive in the brain environment. But when you look into the clinical perspective, it is also not good to maneuver cell functioning. So when you talk about translation should be very much simple. And if, say, for example, you are giving a TPA, you are busting the clots are gone, the circulation is revived. It is a very simple mechanism. But getting that mechanism into picture needs a lot of hard work. So it is only the first, we should know whether the mitochondria is going or not, whether the persistent mitochondria will have some effect. There's again another point, and another study will be required, and that where we may need to go to the genetic aspect, and we have to talk about the personalized medicine.
Because I have seen some results where actually the time-bound movement of mitochondria from cell to cell has been shown in many papers. So I thought you did something like that or not.
I have seen, I know that. And Professor Anurag Agarwal from IB, I think.
IGIB, his data only. Actually, I saw his data.
Yeah, so Dr. Agarwal, what he has taken the lung cells actually. But he has a different aspect of your mechanism, but we are deciphering a different aspect actually.
Okay. I think Imran, you can take over now.
Yeah, please.
Thank you so much, Professor Bhattacharya, for this insightful talk. I'm afraid that we are now at the end of the first day's proceedings, and we have had a series of extremely informative presentations from this diverse field of neurosciences. And I'm sure that all participants will agree with me that it has been a fruitful day after all. So thank you everyone for your time, and we look forward to seeing you tomorrow again. Please join us at 9:30 a.m. Indian Standard Time tomorrow. Thank you very much again, once again, and see you tomorrow.